Hi Manjula, there are many people in the waiting room. I will let people in. Can you just check YouTube live? I have gone live on YouTube just now. Maybe from dispatches.
You started on YouTube, is it? Okay. Um, it's just loading from quite some time. It should show the slide now.
It should show our videos. I can see it on YouTube. Let me check once again.
This is on dispatches.
I'll check on dispatches as well.
Hmm. Refreshing, let's see. Just loading.
I can see it on dispatches also. Maybe there is some internet issue. Should I let people in or should we wait for Karishma to join?
Dispatches, that's working.
Working, no?
Yeah.
Take care.
So maybe we need to wait now until Karishma comes.
Yeah.
How was the last session? I thought it is continuation of that.
Networking session is on another Zoom link, which is going on. I
अच्छा,
left at 4. खत्म हो
अच्छा,
गया? So I think she'll probably join now. Rituja has come. I'm letting her in. I will make Rituja co-host. Hi Rituja.
Hi.
I'm making you the co-host now. Just make sure you let people in, okay? Because there are so many people messaging me separately that we are in the waiting room. Please let us in. And because I'm also doing some other work, then it becomes a problem.
I'm letting in most of the people, but
Yeah,
only
just be
people.
careful with up because sometimes it doesn't pop up. Then they might just be in the waiting room.
Now
And
also,
also,
now
yeah,
also.
now there are, but let's wait for Karishma to join.
Okay.
And also, whenever people join, just mute them, okay? Because there is a lot of background noise that is coming. I'm trying to do it as much as I can also. You also
Yeah.
just keep a note and mute people whenever they join. Hi, Karishma. We are live on YouTube for this session. There are many people in the waiting room. Should I let them in? Or maybe Rituja can, but or should we wait for 1 or 2 minutes? Sandhya Kaushik has also joined.
Yeah, let me know when to let in.
And I forgot to announce the giveaway for the first
Yeah,
session.
I was
So
about to
the
say.
winner is Snehal Patil actually. So I'll announce it now. I see her in the waiting room actually. So remind me, Rutuja, Arushi, if
Yeah,
you
yeah, yeah.
can. There's too much going on actually. Um, Maybe we need to divide this better, say 1 hour or something. Anyway, remind me. ठीक है. Hmm. Rutuja, I think Arushi, if it's okay, shall we start getting people in? Huh?
Yeah,
Okay,
yeah.
I'll let them in.
Hi Deepa, nice to see you again.
Hello.
How are you?
Good. I'm good. How are all of you?
Good.
Hey,
Hi,
Hi
Sandhya.
Sandhya.
Hi, Geeta.
Hi.
Rutuja, there are people in the waiting room, Rutuja. I'm letting them in. Can you just keep an eye?
Actually,
Yeah.
I'm not able to see anyone
No,
in the
just now Shamima entered. Just now Shamima entered. Shamima, hi.
Hello.
Good afternoon.
Good
Shamima, I
afternoon.
know you've shared your slides on the participant slides and I've downloaded it. But would you like to share it yourself?
Yeah, I'll try sharing it.
Sure, sure. There'll be like a brief introduction and then you can share. If you can't share, we can share it. No worries. Huh? Nice to see you early,
Yeah.
up and early. Hi Deepa, hi Sandhya. We will start at 2 with a little introduction and then the talks.
Hi, sure.
Sounds good. And I'll just tell you, we have 6 speakers. So it's an action-packed session. Yeah, it's action-packed. So we'll stick to time. 20 minutes of talk plus 10 minutes Q&A. And if there's any leftover time, we'll take open Q&A at the end.
Are my slides visible?
Not yet, Shamima, because I'm screen sharing.
Oh, okay.
So let me stop share. You try.
Yeah.
The 10 minutes of Q&A is from everybody, right?
Oh, no, from the expert mentors, from both of you. So if there's leftover time, we'll take from the audience. But otherwise, it's largely from both of you. Yeah. Sounds good. I think we're good to go. Shamima, you want to try screen sharing?
Yeah, I've started. Are the slides visible?
Yes,
Yeah,
Yes,
I
I
can
can
yes,
see
the
see.
them.
slides are visible.
Great. So you can share, Shamima. Huh? Now
Okay.
I will share as we'll start at 2 p.m. But you share when you want to give your talk. Sounds good?
Yeah.
Okay. Okay. So you stop sharing now. Now I will share and then.
Okay.
Hmm. We had a very good turnout for the morning also. Yeah.
This is just run the next 3 or 4 days. This week is fully this, is it?
This is this. Yeah. Yeah.
So you have around 50 speakers over the whole
50
week?
speakers over the whole week, Deepa. And the 10 themes were curated based on the Company of Biology's journal themes.
I see.
So there are some omissions like computational biology, biophysics, and so on. But we had to align with the journal themes, to get the grant. But we tried to fit them in elsewhere as much as we could.
Sounds good.
Okay, we'll start at 2 p.m. All right.
Yeah.
Okay, it's 2 p.m. Welcome all. Good afternoon. Postprandial dip in blood sugar levels. Hoping not. Hoping not. Hope you had a good lunch and there's no dip. With the science should keep you excited. So welcome all to the second session on the first day of PhD Cafe. And our segment today afternoon is Cellular Biology. PhD Cafe is supported by Company of Biologists to India Bioscience. And we've curated a week-long science fest, starting with today morning, which was on plant biology. And the seeds have taken uh, root, and now we are proceeding to cellular biology. Um, it's going to be a very exciting lineup of talks, starting with Shamima, then Kalyani, Saujanya, Shreyas, Sanjana, and Prajna Dipta. Thank you all for being here. Um, your interest in PhD Cafe was quite unprecedented. We curated this list of cellular biology speakers from a very vast pool of of, of candidates who wanted to speak. So congratulations and welcome. This is how it will flow. 20 minutes of speaker time, 10 minutes of Q&A from our expert mentors. And if we have time for audience Q&A, we are happy to take that. But the audience can always interact through the chat window. Um, so make sure you can see and hear us. Use the chat window to type in your questions. Our session in the morning was very interactive in the chat window. And PhD speakers, if you don't get a chance to answer the audience question after your talk, you can answer in the chat window. You can take it off sort of offline there. Okay, so we'll have expert mentor feedback after every 20-minute talk. Do stick to the time. My colleague Rutuja here will put a alarm icon that tells you your 20 minutes is up. So that's time to start wrapping up. Okay. And of course, in the spirit of a scientific talk, feedback is welcome, but it should be constructive to make the work better. And I'm sure we'll adhere to that. These are our expert mentors, Sandhya and Deepa. Thank you for being here. Thanks a ton. And we look forward to this afternoon of science with you both. Now, I have built up this giveaway, but guess what? I forgot to announce the winner in the morning session. So apologies. There was probably too much going on. But the winner for today's morning session was Snehal Patil, who I believe is here in the afternoon session as well. So thank you for keeping the chat window very engaging, Snehal. You are our audience winner. And all of you who are here for the cellular biology session, we have a second book giveaway today afternoon. And what you need to do for that, to get a physical copy of this compendium on challenges and opportunities in doing a PhD in life sciences in India, is to keep the chat window active. Ask questions, give suggestions, seek feedback on your talk, and give feedback to other people's talks. So keep the chat window active and you may be in line for the second giveaway. So congratulations, Snehal, and one more book to be won this afternoon. And so, like I said, if we have time, we'll do the open Q&A. But with that, I think we are ready to get started. So Shamima, thanks for being here. I will stop sharing now and you can start sharing. And once you start sharing and speaking, we will start your 20-minute timer. Great. Welcome.
Are the slides visible?
Yes, Shamima, it's visible. Rutuja, start the 20 minutes. Yeah.
Yeah. So good afternoon, everyone. My name is Shahima Khatun, and I am doing my PhD under the supervision of Dr. Sabyasachi Sanyal from Biochemistry and Structural Biology Division in CSIR CDRI Lucknow. So the project I'm working on is dissecting the role of ACG, an adipo-R agonist, and elucidating its protective role in MASLD or MASH. So metabolic dysfunction associated steatotic liver disease is a chronic liver disease, and it is a very common liver disease nowadays worldwide, as it affects 1 in 4 adults. And it is always accompanied by metabolic syndromes and its components like obesity, insulin resistance, diabetes, and dyslipidemia. So mainly my work focuses on adiponectin signaling pathway. So adiponectin is an adipocytokine, an anti-inflammatory adipocytokine that exerts numerous beneficial effects via its primary receptors, adiponectin receptor 1 and 2. And depletion of adiponectin has been shown and seen that it hosts a lot of metabolic disorders, including MASLD. So decrease in adiponectin levels is associated with metabolic syndrome and cardiovascular diseases and obesity. And in liver, adiponectin helps in glucose and lipid metabolism and exerts insulin sensitizing effect. And so adiponectin supplementation therapy is seen as an treatment option for this, but due to its adiponectin's relatively large size, that is it ranges from 90 to 180 kilodalton, as it is present in various forms, and it's complex oligomerization, and it is present in various forms like in trimers or hexamers. So it is very difficult to use it as a supplementation therapy. Therefore, a small molecule adiponectin mimetics may provide a unique opportunity for MASLD and associated metabolic disorders. So the underlying cause of hepatic steatosis is said to be sedentary lifestyle, insulin resistance, obesity, and diabetes, as I've mentioned earlier. And if we explain steatosis, it is simply the infiltration of fat in the liver cells, and it disturbs the metabolism of these cells. So the mandatory feature of MASLD is presence of liver steatosis, that is, if there is inflammation and in the absence of other chronic liver diseases. Now, the inflammation leads to the dysfunction in the carbohydrates and lipid metabolism. That is, the fatty acid synthesis increases and fatty acid oxidation decreases. Along with all of this, there is mitochondrial dysfunction, chronic oxidative stress, and other factors. And along all of these factors makes MASLD and MASH, that is the more severe form of steatotic liver disease, that is metabolic dysfunction associated steatohepatitis, most common clinical pathological conditions. Over here, I'm showing that how the normal or the healthy liver proceeds to the end stage of liver disease, that is cirrhosis or hepatocellular carcinoma. That is, it starts with merely formation of fat vesicles and disturbance in the metabolism. Later on, it proceeds to the infiltration of inflammatory cytokines, and then scarring and fibrosis, and ultimately leading to cirrhosis. Now, adiponectin signaling in a healthy liver helps the liver in decreasing oxidative stress, inflammation, gluconeogenesis, and lipid synthesis, along with improving insulin sensitivity and fatty acid oxidation. Now, adiponectin is a fat derived, as I have already mentioned, and it binds to two specific receptors, adiponectin R1 and R2, which belong to the 7 transmembrane domain receptor family, but they are not coupled with G protein. Now, R1 is ubiquitously produced with highest expression in skeletal muscles, whereas R2 is predominantly seen to be expressed in the liver. Now, these receptors plays crucial role in glucose or lipid metabolism, inflammation, mitochondrial biogenesis and its function, chronic oxidative stress, and therefore it has a huge therapeutic potential. Now, relevance of my study is that the therapeutic potential of adiponectin has been described in more than 12,000 publications till date, and translation of adiponectin, as said earlier, is a difficult task. So over here we have used a small molecule to regulate these receptors. Now, this gives it has given us an opportunity to explore this field, and ACG, a small molecule activator of adiponectin, was identified in our lab. And ACG has been shown to help with hepatic carcinoma and improve liver health in some unrelated studies, although its effect in MASLD has not been studied elsewhere, and its mechanism is still unknown. So over here, we have done like ACG, we have proven that ACG is a selective adipo-R2 agonist. Now, ACG is a epigenin 6C glucoside, also known as isovitexin. It is isolated from a plant, like it is a plant, comes from a plant sources. Now, over here, we have taken HEK293 cells, which are known to have lower endogenous adipo-R1 and R2 receptors. So we have overexpressed adipo-R1 and R2 in HEK293 cells, and we have treated it with ACG and adiporon. Adiporon is a dual agonist of adipo-R1 and R2 receptors. And ACG, which we are trying to which we are trying to prove over here, that it works through adipo-R2 receptor only, or has the higher affinity to adipo-R2 receptors. So from this experiments, it was seen that adipo-R2 overexpressing cells were able to increase the luciferase activity. Now, this luciferase activity comes from PGC1 alpha Luc plasmid that is also being transfected along with adipo-R1 and R2. And this PGC1 alpha is the main target of adiponectin receptors, adiponectin signaling. So adipo-R2 transfected cells had increased luciferase activity, suggesting that this ACG works through R2 receptors, whereas adiporon, which is a dual agonist, it worked with both the adipo-R1 and R2 transfected cells. Now, the A-glycan form of ACG, epigenin, was also used to test whether the changes in the structure causes any disruption in the activities. And it was seen that the luciferase activity was not much in adipo-R1 and R2 transfected cells treated with epigenin. Now further we checked that HEK293 cells transfected with ADPO-R1 and R2 treated with ACG. AMPK and P38 are also the downstream signaling of this receptor, and ACG was shown to increase the activation of AMPK and P38, that is via their phosphorylation. And again, with the setup of same as the figure 1A, we calculated the EC50 of ACG, and we saw that ACG was working through ADPO-R2 and had an EC50 of 384 picomolar, whereas with ADPO-R1 it had 6.9 micromolar of EC50, suggesting that it was 10,000 times more preferring ADPO-R2 over ADPO-R1. Now, as we know that ADPO-R2 is predominantly expressed in liver, we next checked the activity. We next checked the effect of ACG on liver cell lines, that is HepG2, and we saw that treatment with ACG increased the phosphorylation of AMPK and P38 as well as AKT. And we also again checked HepG2 treated cells with both ACG and epigenin, and saw that ACG was increasing the phosphorylation of P38 at 100 picomolar dose, whereas for epigenin, it started from 1 micromolar dose. Later on, we also checked PGC1 alpha and PPR alpha expressions in HepG2 cells treated with ACG, and we saw that there was an increased expression of PGC1 alpha and PPR alpha. Now, these ADPO-R2 rich HepG2 cells were treated with ACG and checked for the signaling markers or the proteins involved in it. And depletion of this ADPO-R1 and R2 receptor did stop the effect of ACG, but only with ADPO-R2. Whereas ADPO-R1 depletion didn't have much of the blockage of ACG. So it suggests that again that it works through ACG.
Shamima, sorry to interrupt, but could you please use the pointer to show whatever you're talking about?
Okay.
Yeah,
Sorry.
thanks.
Yeah, so over here we have developed an in vitro model of fatty liver, and like only the fat accumulation part, not the infiltration of inflammatory cytokine, which was not possible over here. So we have done oil red O staining to check the lipid accumulation, and we have treated the cells with 2 treatment regimen. That is one is co-treatment, in which the palmitate, which was used as a fat source or the lipid source, palmitate along with ACG for 24 hours, and the next treatment was palmitate for 24 hours, and then treatment with ACG along with palmitate for next 24 hours. That was known as that we called as therapeutic treatment. And we checked the lipid accumulation, cytotoxicity, and the inflammation marker P65 NF-kappa B, and we saw that ACG could ameliorate the effect of palmitate in these cells. We also checked for the mRNA expressions of various markers of inflammation, fatty acid synthesis, oxidation, fatty acid oxidations, and we also checked the main downstream target of adiponectin signaling pathway, that is PGC1 alpha. And over here, we also did the mitostress experiment to check the mitochondrial health of the cells treated with palmitate, and again, like induced lipotoxicity induced in these cells with palmitate and then treated with ACG. And we saw that ACG could increase the oxygen consumption rate, maximal respiration, and ATP production. And it also improved the fatty acid oxidation of these cells. In this fatty acid oxidation experiment, these cells were glucose deprived, and only source for them to use for increasing their oxygen consumption was palmitate. And in this model, we saw that there was an increase in fatty acid oxidation, which was actually dampened by palmitate treatment. Next, we did we again checked for the various markers and the inflammation marker. We checked for the main target of adiponectin signaling, PGC1 alpha. So over here, we found a little bit of overlapping activity of both the receptors. So the depletion of both the receptors, because this treatment time is for 24 hours, both the receptors lost the activity of ACG, and over here again, the mRNA levels of various markers were seen, and we saw that adipo-R1-R2 depletion lost the effect of ACG over here. Whereas adipo-R2 depletion over here, we have checked the lipid accumulation, and we saw that adipo-R2 depletion blocked the effect of ACG, suggesting that again that it works through the adipo-R2 receptor. And over here, we have done ROS accumulation with DHE staining. And next, we wanted to know that which signaling pathways actually it is going through. So we used AMPK, P38 MAP kinase, and PI3 AKT inhibitors, and we blocked the and we checked for the blockage of ACG mediated amelioration of palmitate induced induction of these NF-kappa B expression, lipid accumulation, and ROS generation in HepG2 cells, and we saw that inhibition of AMPK and P38 actually blocked the effect of ACG, and so this suggests that it works through the AMPK and P38 pathway, like the downstream pathways of adipo-R2. Now to mimic the fibrosis-like condition, we have used stellate cells over here. These stellate cells were also induced by palmitate, and their activation was induced by palmitate. And then when ACG treatment was given to them, there was a decrease in the fibrotic markers like alpha SMA and TIM2 and collagen 1. Next, this whole in vitro study was replicated in vivo, in which we have used an ApoE knockout mice model. So ApoE knockout mice model actually mimics the fatty liver, like MASLD like condition, and over here the mice are fed with high fat and high fructose diet. These are some of the parameters that were checked, that is body weight, serum, and hepatic parameters. So over here the body weight was there was no difference in the body weight seen. There was an increase in body weight in the HFRD fed, the normal chow fed mice, whereas HFRD fed mice were losing weight. Not losing weight exactly, but there was a decrease in weight. And all the liver enzymes, there was no change or significance seen, whereas triglycerides in the liver, like the serum parameters of triglycerides were seen, that silymarin in elevated. So over here we're using silymarin, which is a therapeutic control, and in vitro studies we have used ediperon, that is the pathway comparator. Now the liver triglyceride. So serum triglyceride didn't show any significant change. There was an increase as compared to HFRD, while the triglycerides from the liver showed that ACG treatment decreased the liver triglycerides inside the liver, and also silymarin also decreased the liver triglycerides in the liver. We have also done an ultrasound imaging to visualize the lipid deposition, and we saw that as compared to baseline, there was a decrease in the lipid deposition in the mice treated with ACG and silymarin as compared to HFRD-fed mice. Now, this normal chow diet mice also showed lipid deposition, which you can see with this white things. So that is because the mice were ApoE knockout mice, and they are dyslipidemic mice. So that's why there was this abnormality seen in the normal chow diet. Now we have seen the hepatic histologies, that is, we have seen HNE staining, we have seen the liver tissue, and we have also done picrocereus red staining for fibrosis, and we have quantified it. And for scoring, we have used Kleiner method, and we saw that there was a decrease in the score, NAS score, and there was decrease in fibrosis in ACG and Cellularine treated mice. Over here, we have again checked for the lipid deposition with oil red O stain, and we saw that ACG and Cellularine had reduced lipid deposition as compared to high fat slash fructose diet mice. We over here we also checked for glycogen, hepatic glycogen, and we saw that the disruption which was caused by HFRD when treated with ACG and Cellularine, it was ameliorated. We checked for the F4 by 80 positive cells, that is an inflammation marker of M1 macrophage, and we saw that there was a decrease in M1 macrophage content in ACG and Cellularine treated mice. And we also did the Western blotting of these tissue samples, and we found that there was a decrease in inflammation marker, that is P65 NF-kappa B, an increase in PGC1 alpha, decrease in alpha SMA, that is a fibrotic marker, and increase in PPR alpha. And we also saw the again with the help of the liver tissue, we have done the RT-PCR of the samples, and we saw for inflammation, fibrosis, and fatty acid metabolism. And we have also done the lipid peroxidation, and this was done again with the liver tissues. And over here, we also checked for the phosphorylation of AMPK and AKT and P38. And we saw that there was an increase in phosphorylation of AMPK and P38 in the animals treated with ACG and Cellularine. Now, the second model of that we used was CDHFD. So CDHFD is a choline deficient L-amino acid, high fat diet induced hepatic, like a hepatic steatosis model. So in this C57BL6 mice were fed with this diet, and then we have assessed the fed with this diet and given the treatment of ACG and Cellularine, and then we have assessed the same parameters as we assessed in ApoE knockout mice, like the body weight, liver enzymes, and fasting glucose, and liver index, that is liver weight by body weight. So over here in this, the normal control mice is like a normal control mice, and the other ones are fed with CDHFD. So there's a difference between their like the histopathology. You can see there's a difference. There is fat vesicles formed in the liver tissues, and there is infiltration of inflammatory cytokines. And over here, there is an increase in weight of normal chow diet fed mice, and the weight of the animals fed with CDHFD, there isn't any change. They are consistent for all the treatments plus CDHFD fed alone. We have again done for picrocereus for fibrosis and F4 by
Shamima, we can't hear you. I think you've got muted. Also, Shamima, we should wrap up because 20 minutes is up. We'll
Okay.
head to the 10-minute Q&A now. So you want to wrap up quickly, maybe?
Yeah, so these are the models that I have used, the in vitro models, in vivo models, and the methods, that is luciferase reporter assays, Western blotting, and all of this. Finally concluding that ACG, it binds to adiponectin receptor 2, whereas there is some overlapping with adiponectin receptor 1, which we have to still study and find. Which reduces inflammation, fatty acid synthesis, increases fatty acid oxidation, that in a whole decreases lipid accumulation and blocks fibrosis, that is maintaining a healthy liver. I would like to acknowledge the CDRI intramural funding and our collaborators in CDRI and Zydus Research Center. And I would also like to thank India Bioscience for providing with this opportunity. Thank you.
Thank you, Shamima. That was a lot of data. That was a lot of data. All right, I'll hand over to the expert mentors for their suggestions and feedback for you.
Okay, so couple of quick questions. So you showed some data which talked about. Can you hear me,
Yeah,
Shamim?
yeah, yes,
Okay.
ma'am.
You showed some data which suggests that there was a change in phosphorylation of AMPK and AKT with the application of ACG. Did I get that correct?
Yes, yes. In HEK 293 cells,
Yeah,
overexpressed with adipo-R1 and R2. Yeah.
yeah. So my question was, why was this phosphorylation relevant? And I also saw that these effects were somewhat different when you just had adiponectin as well. So the response in some cases seemed to be higher with the treatment of this agonist rather than the cytokine itself. So do you have any comment
No,
on?
so the control that I'm using is a synthetic form, and that is adiporon. That is not adiponectin. So adiporon is a dual agonist of R1 and R2, and it was the group from Japan, Kawasaki. They proposed this, and it works in higher concentration, and it shows some cytotoxicity. That's why they have not proceeded with this as a treatment option. So over here, we are using it as a control. So it is not adiponectin. It is adiporon. So it has some cytotoxic effects as well.
So you think that these differences between ACG and
And
this,
adiporon, yeah. And we are using it in a very high concentration, that is 5 micromolar.
what is the relevance of the phosphorylation of AMPK and AKT?
So phosphorylation of AMPK, basically AMPK is important because it is directly, it comes downstream of the adiponectin receptor 2. So when the adiponectin, if we see in a
What do
normal
you
liver,
mean? Sorry, can you just break that down? When you say it's directly downstream, so what happens downstream of adiponectin receptor, that the receptor turns on some signaling cascade?
yeah, so it first dimerizes APPL1. So when APPL1 dimerizes, it activates liver kinase B1, which further activates AMPK by phosphorylating it at threonine 172 site. So that's why it is important, because it further then activates the PGC1 alpha and all the other signaling pathway that is important for the liver health.
So without the phosphorylation of AMPK, PGC1 alpha, and all of that doesn't get, I don't know what, phosphorylated, turned down. I mean, I don't understand. Can you just explain to me what the link between the phosphorylated AMPK and the downstream effects that
So
you're
the phosphorylation of AMPK means it gets activated and it further activates the downstream signaling. That is the activation of
AMPK
PGC1.
without phosphorylation is not activated.
Yeah.
It's not active at all.
It is not activated.
I see. Okay. Deepa, sorry.
So I was just wondering, what are the long-term effects of treatments of this with this chemical, with ACG?
With ACG.
Because with you in your cells, I think you've done a 24-hour treatment. It wasn't clear to me how long you were administering this to the animals. Because presumably for this to have some effect, it has to be taken for a while, for a number of
So
days.
the two animal models, So the two animal models that we have used for ApoE knockout mice, we have given it for like 7 weeks. Like 7 weeks with the only the high fructose, high fat diet, and then further 7 weeks high fructose diet plus ACG treatment. And for the other, it was 8 weeks CDHFD.
And so are there, did you look at other organs, for instance, to see if there are any off-target effects? Because you're giving it at clearly a high concentration also.
Yeah, no, we have not checked other organs, but our lab works on liver and muscles. So we are working on muscles right now. After this, we are checking for muscles, like what the effects are.
No, also because it affects pathways which are fairly commonly used across different cell types and in different cellular scenarios. So I was just wondering, especially, if you have AMPK going up and down to these levels, you're going to be affecting a lot of things in a lot of cells. So what sort of is the long-term implication in an animal when you feed it with such high levels of this drug? Do you have any insights or thoughts on that?
So, like we started this study, but as we are using the concentration that we are using is fairly less than the concentrations that I have seen in the publications and papers like literature. So, like they have used in 5 micromolar concentration that, but the Cmax in mice is 297 nanomolar. So over here we are using like 594 nanomolar in mice that we are using. So that is one thing, and also we were actually like using, we want to use this as a like like a backbone for researchers to further find something that is related to adipo R2, like agonists for adipo R2, a better one, or which would be like in lower concentration but has effect. Like we are trying to say that there is this signaling pathway also that we can play with to like help with MASLD.
Can I ask one more tiny question?
Yeah.
So I was just curious, you don't see these changes in this AL2, ALT, SGPT, and
Like
AST,
liver
SGOT.
enzymes. Yeah.
And this is usually, your standard readout for liver health. So I was
Yeah.
just wondering, you see changes in the triglyceride levels, etc., but not in these. So why is that the case?
Yeah, so that was like something, it didn't work according to the normal physiology, or the main things that we actually see when we test for fatty liver, the main enzymes, but we didn't see any change. So we are also working on that. We are also checking for what is actually the reason behind, there isn't any change in the liver enzymes. As we can see, there isn't much change in the triglyceride in the serum levels, but when we are checking it from the hepatic, like from the tissues, there is changes in the triglycerides.
Thank you.
Can I ask one last question?
Yes.
So metabolic syndrome, which one of the problems with metabolic syndrome is the steatotic liver disease. Are you suggesting that ACG will act purely at the liver, or
Yes.
can it have beneficial effects
In the metabolic.
in just metabolic syndrome generally through tissue level interactions or signal cascades?
Yeah, we
Is that
haven't.
something that, because you have looked at a large number of blood parameters, and I was not, or whatever, the serum parameters, right? You looked at it in your mouse model. So it was not clear to me how many of those parameters arise only from changes in the liver as opposed to have contributions from other tissues.
So like triglycerides and everything that is over here, finally we conclude that it basically only works with the liver, not in general, like for the metabolic syndrome.
I see. Okay.
Okay. Thank you, Sandhya, Deepa. Thank you, Shamima. That was an in-depth analysis. Sandhya and Deepa, thank you so much. Shamima, thanks a lot.
Thank you,
It
ma'am.
was a wonderful talk. Hope you gained something out of it. And Snehal has a question in the chat window, which you
Yeah,
can
sure.
respond to in the chat window. Okay, that kickstarts cellular biology with session 1, talk 1. And next speaker is Kalyani. So Shamima, you can stop sharing and Kalyani will start sharing. And yeah, you just let us know, Kalyani, when you're ready to share your slides. Kalyani is going to talk about, it's an interesting title, Unlocking the Secrets of the Cap, the role of RNA capping enzyme triphosphatase in Toxoplasma gondii, survival and stability. Wow, that sounds very exciting, Kalyani. Thank you. Go ahead. We can see your slides. Excellent. Go ahead. So you have 20 minutes plus 10 minutes for Q&A. Kalyani, we cannot hear you. We cannot hear you, Kalyani. You want to unmute, please? Yes, Kalyani, can you say something maybe so we can check? Kalyani, we cannot hear you. Rutuja, maybe you can check what the issue is.
Yeah, I will try contacting him.
Yeah. Kalyani, I don't know why there's a green arrow next to her name. We'll just give it a minute or two.
It's because
Uh,
she's sharing her screen. That is why the
okay,
green.
okay, that explains that. But Kalyani, can you say something maybe? We cannot hear her, unfortunately. All right, I think we will move on if we can, and Kalyani will come back to you at some point. This looks like some technical issue at your end. So I think our next speaker is Saujanya. Saujanya, are you here?
Yes, I'm here.
Can you please, yeah, thank you for being there and for giving your talk half an hour earlier. But
Yeah.
Kalani, maybe you stop screen sharing and Saujanya can screen share. Sure.
I hope I'm audible to everyone. Hello? Hello?
Yes, you
Are
are.
audible.
Sure. Hi, everyone. Welcome you all to the PhD Cafe seminar series. And first of all, thanks to India Bioscience team for giving me the opportunity and great platform. I am Saujanya Vinayagamurthy from CSIR Institute of Genomics and Integrative Biology. With the guidance of Dr. Shantanu Chaudhary, so far I've been trying to understand the extratelomeric impact of TRF2 in neural stemness. I'll begin from the ends of the chromosomes. Let me just turn on the pointer, which are telomeres. The major function of telomeres is to protect the chromosomes. Telomeres are enriched with this TTA-GGG repeat sequences. In 1980s, it was first shown that these G-rich sequences can undergo Hooke-Steen base pairing and form DNA secondary structures called G-quadruplexes. For a very long time, researchers thought that these G4 forming structures reside only at the telomeres, but later researchers started noting elsewhere as well. G-quadruplex forming structures are non-randomly distributed and highly enriched in the promoter regions of the genome. A gene ontology analysis was done from this group. And what was interesting for us to see is that the genes that are involved in neurogenesis and cell differentiation had significantly higher frequency of promoter potential quadruplex forming sequences. Now coming back to the telomeres are capped by this multiprotein complex called the shelterins. The protein of our interest is telomere repeat binding factor 2, TRF2. Why? Because similar to G4 structures, TRF2 also occurs outside the telomeres. It functions as a transcriptional regulator. Importantly, the work from our group has shown that TRF2 binds to the promoter quadruplexes for the transcription function. So one such case study where TRF2 functions as a transcriptional regulator is the human telomerase reverse transcriptase gene, where TRF2 binds to the promoter quadruplexes, recruits REST, recruits PRC2 complex, and deposits H3K27 ME3 mark on the promoters for gene repression. Now, what is REST? REST is RE1 silencing transcription factor, otherwise known as neuronal restrictive silencing factor. It has several functions. One is to maintain the embryonic stem cell characteristics. It represses the neuronal differentiation genes, and it represses the neuronal differentiation genes in other cell types as well. Also, it is a tumor suppressor. So far, what we know is that promoters of differentiation genes contain quadruplex forming sequences. TRF2 binds to the promoter quadruplexes, and TRF2 can recruit REST. Connecting these dots, we derived a hypothesis that if the promoters of differentiation genes contain quadruplex forming sequences, they can also possibly be regulated by TRF2 mediated REST recruitment. To understand this hypothesis, we chose two models. One is SHSY5Y, which is a human neuroblastoma cell line. These cells, which when treated with retinoic acid and BDNF, can undergo neuronal differentiation, which is marked by increase in neurite length and decrease in the soma. and this is widely used in the field of neurobiology to understand neuronal differentiation. Our second model is the mouse neural stem cell model. To understand the loss of function of TRF2 in mouse neural stem cells, we created a conditional inducible knockout model of TRF2, wherein the Cre recombinase expression is driven by the nestin promoter and the TRF2 is floxed. These mice, which when treated with tamoxifen, expresses Cre recombinase and knocks out TRF2 in mouse neural stem cells. And nestin is highly expressed in mouse neural stem cells, so it is conditionally deleted only in the mouse neural stem cells. From this mice model, we derived hippocampus and then we cultured them in vitro as neurospheres, which are enriched with the mouse neural stem cells. And this is characterized by the expression of PAX6, which is a neural stem cell marker. With this model systems in our hand, we first wanted to understand if TRF2 can transcriptionally regulate neuronal differentiation genes. For this, we utilized chromatin immunoprecipitation assay. From now on, I'll be referring to this as CHIP assay. Though this is from a different cell line, previously our lab had done a TRF2 CHIP-seq and a gene ontology analysis was done. And what was peculiar for us to see is that those genes which were involved in neuronal differentiation, such as those genes which are involved in synapse formation and neurotransmitter release, were significantly enriched for TRF2 binding. From this data set, we selected few genes such as APP, BDNF, GFRA, L1CAM, MAP2, and presenilin, those that are involved in neuronal differentiation. In this gene set, we also confirmed that these genes have potential promoter quadruplex forming sequences through bioinformatic analysis. With this gene list, we first question if TRF2 can bind to the promoters of these neuronal differentiation genes in the neural cells. So we did CHIP assay, and then we noticed that both in SHSY5I and in mouse neural stem cells, we could see that TRF2 bind to the promoters of the neuronal differentiation genes. Now that TRF2 is binding to the promoters, we wanted to understand if perturbing the TRF2 levels can alter the gene expression of the neuronal differentiation genes. So basic domain, dimerization domain, and MIP domain are the regions that are the domains of TRF2. And basic domain and MIP domain are the regions that are involved in DNA binding. The deletion of these domains causes the dominant negative mutant form of TRF2. These constructs were overexpressed in SHSY5I and mouse neural stem cells. And what we noticed that wild-type overexpression did not alter the expression, whereas the dominant negative mutant form of TRF2, which when overexpressed, led to the activation of those genes which had promoter TRF2 occupancy. This tells us that the DNA binding activity of TRF2 is essential for TRF2 mediated repression. On the other hand, when we silenced TRF2 in SHSY5I cell line, we saw that with increasing time points, there was increase in expression of those genes which had promoter TRF2 occupancy. Similarly, in the mouse neural stem cell model, when TRF2 was knocked out, we could see the elevated expression of those neuronal differentiation genes. This tells us that loss of TRF2 increases differentiation gene expression. Now that we know that with loss of TRF2, there is elevated increase in the neuronal differentiation genes, we wanted to understand if this elevated gene expression translates to a cellular phenotype. So for this, we did immunofluorescence assay. And then this is how the normal SHSY5I cells look, which is marked by beta-3 tubulin, and TRF2 is marked in green here. And when wild-type TRF2 is overexpressed, we could see that there is no change in the cellular phenotype. But when the dominant negative mutant form of TRF2, that is delta B delta M, which when overexpressed led to a neuronal differentiation phenotype. Similarly, when the TRF2 was silenced, we could see that there was increase in neuronal differentiation phenotype in SHSY5I cells. The same question we wanted to test in mouse neural stem cell model. For this, we utilized neurosphere formation assay. The number and size of neurospheres that are formed is directly proportional to the stemness and proliferative potential of these cells. So this is how the normal neurospheres look, which when treated with vehicle control, they are nice and intact. When these cells were treated with tamoxifen, which creates knockout of TRF2, there was compromised ability to form neurospheres, indicating that there is a reduction in the stemness property. To further substantiate this, we quantified the expression of Ki67 through flow cytometry. And then we noticed that with reduction of TRF2 levels, the proliferative potential decreases. And we questioned if it increases the differentiation property. And we quantified MAP2, which is a neuronal differentiation marker. With loss of TRF2, we could see that MAP2's expression increased. This tells us that loss of TRF2 causes differentiation. Now that we know that loss of TRF2 causes neuronal differentiation, we wanted to understand the levels of TRF2 during normal neuronal differentiation. For this, we differentiated SHSY5 cells, and then we noticed that upon differentiation, there is drastic reduction in the decrease of TRF2 levels. And we also performed chip assay. We noticed that upon differentiation, the promoter occupancy of TRF2 on the differentiation genes also decreased. This tells us that neuronal differentiation is accompanied by the loss of TRF2. So the summary so far is that TRF2 binds to the promoters of differentiation genes to keep them repressed and to maintain the stem state. And loss of TRF2 leads to the activation of these genes and causes a differentiation phenotype. Next, we wanted to understand the TRF2 dependent repressor complex that is being brought to the promoters for the gene repression function. We know from the literature that EZH2, which is the catalytically active component of PRC2, is highly involved in keeping the neuronal differentiation genes repressed. And then we also knew from literature that REST is the molecule that recruits is PRC2 to the promoters. With this knowledge, we question that if REST can interact with TRF2 or not. So we did co-immunoprecipitation assay, and then we saw that TRF2 could interact with REST. Next, we wanted to understand if TRF2 can recruit REST to the promoters. For this, we silenced TRF2 and performed a chip for REST. And then we noticed that with loss of TRF2, there was reduction in the occupancy of REST on the promoters. On the other hand, we silenced REST and performed a chip for TRF2. And then we noticed that with REST loss, there was no change in the occupancy of TRF2 on the promoters. This tells us that TRF2 is the molecule that recruits REST to the promoters. As I mentioned earlier, EZH2 is recruited by REST. We wanted to understand if EZH2 occupancy on the promoter is TRF2 dependent or not. For this, we silenced TRF2, and then we noticed that with loss of TRF2, EZH2 occupancy on the promoters reduced. This tells us that EZH2 occupancy on the promoter is TRF2 dependent. The final readout, which is the H3K27ME3 repressive mark on the promoters, we wanted to test if it is TRF2 dependent or not. And then we noticed that with loss of TRF2, 27ME3 mark on the promoters also reduced drastically. So the summary is for this part is that REST recruits PRC2, deposits 27ME3 mark on the promoters to keep the neuronal differentiation genes repressed. We further wanted to understand if any post-translation modification of TRF2 is crucial for maintaining the neural stemness. So these are the commonly studied post-translation modifications of TRF2. We wanted to understand the influence of this PTMs by generating mutants such that there is loss of function of this PTM. These constructs were overexpressed in SHSY5 cells. And then we noticed that upon overexpression of one particular PTM mutant, which is TRF2K176R, where lysine is substituted with arginine at the 176 position, we could see that there was increase in the neuronal differentiation phenotype. And further, we quantified the gene expression analysis, and we noticed that in the presence of TRF2K176R, these genes overexpressed. Then with this thing, we questioned that why is there a loss of function? We question if there is a compromised DNA binding ability of this mutant, These mutants are flag tagged, and we performed a chip with flag antibody. We could see that this mutant could still bind to the promoters of the neuronal differentiation genes. This tells us that the DNA binding ability is still intact. Now that the DNA binding ability is intact, we questioned if this mutant's interaction with the epigenetic complex is that being compromised. So we did co-immunoprecipitation assay, and then we noticed that wild type TRF2 could interact with REST, whereas the mutant form, which is K176R, could not interact with REST. Next, to further substantiate this, we performed a chip assay in the presence of TRF2K176R. We noticed that REST occupancy as well as the 27Me3 occupancy on the promoter decreased when there was TRF2K176R overexpression. So what does this tell us? That TRF2 has to be acetylated at the K176 residue for it to interact with REST, and loss of acetylation leads to loss of interaction with REST. So the overall summary from our work is that in order to maintain the stem state, TRF2 binds to the promoters of differentiation genes, recruits REST, recruits PRC2 complex, and AZH2 deposits H3K27Me3 mark on the promoters of differentiation genes to keep them repressed. And loss of TRF2 leads to loss of epigenetic complex and the repressive mark, thereby activating these genes and causing a neuronal differentiation phenotype. Thus, TRF2 is indispensable to maintain neural stemness. With this, I would like to thank my lab mates, Samit Kumar Bhatt and Sulochana Bagri, who had greatly contributed to our work, and my PI, Dr. Shantanu Chaudhary, and IGAB and CSAR and India Bioscience team for this opportunity. Thank you, everyone. Now I can take questions.
Can you go back to the model slide?
Yes. Yes, ma'am.
You're not sharing it.
Okay. Is it visible now?
No, it's not. It's not visible.
Just a second.
Saujanya.
Okay. Let me try resharing it.
Sounds good. But that was a very impressive talk, as Snehal has said in the chat window.
Yes.
A very nice flow, very nice logic,
Yes.
even for someone not from the field to follow. So well done. Yes, now I think we're getting there. Yes, we can see your
Yeah.
slide.
Hello?
Yes,
ma'am.
Is your model slide? I don't
Yes,
see.
ma'am.
I think Sandhya means the final one, where you're
Yeah,
talking about the
final-
molecular.
Oh, bundle. Oh, sorry.
Oh, yeah, exactly.
Yes.
This one. So I had a question about your dominant negative construct that you used. What is that? Do you show that does not recruit REST? Is that the reason why it's a I mean, I was not able to understand how
So
it
the dominant negative mutant form cannot bind to DNA. That
Okay.
leads to derepression.
And
So
so
the DNA binding domains are deleted in that mutant. That's how it functions as a dominant negative mutant.
So the recruitment of REST is essential for the continued repression.
Yes, yes.
So the dominant negative version, despite not recruiting to DNA, continue to recruit REST. I mean, I don't understand how that dominant negative works, especially after your model came up. I'm very confused. So some elucidation would be greatly appreciated.
Okay, so the dominant negative mutant form, which I expressed in my first slide, that lacks the DNA binding domains.
Yeah, so it doesn't get on there at all to do
Yes,
any.
yes. So if it doesn't get on at all, REST cannot at all bind to the promoters.
But when REST binds, that's when the repression continues, right?
Yes.
Or okay.
Uh-huh.
If rest does not bind, does it still gene expression occur from those loci?
If rest does not bind, then it leads to de-repression.
Which is basically means you have expression occur.
Huh.
So your dominant negative essentially is going to turn on all your neuronal markers.
Yes, yes.
Okay. Okay. Thank you. I just was not clear what was
Okay, okay, okay.
So I was just wondering, the neurospheres that lack TRF2,
Yes, ma'am.
or rather those cells don't form neurospheres, right?
Right, right.
So what do you think is happening there? Is it more a question of apoptosis or just lack of proliferation or an adhesion phenotype?
Yeah. So it is not lack of apoptosis that we have checked through tunnel
As in,
assays
it's
and
not
fluorescence.
apoptosis. Okay.
It's not apoptosis. So these lack stemness and then they prime to differentiation. That's why there is a loss of stemness. They undergo differentiation.
No. My question is, you don't see any neurospheres forming at all.
Yeah.
So is that, if that's not apoptosis, is that, so you're saying that there are absolutely no stem cells?
There is reduction in the stem cell pool. And these
So,
cells
so I think
spontaneously
my
differentiate.
confusion was more at which stage you're treating with docs.
It's tamoxifen based.
Sorry, tamoxifen.
Yes.
So you're doing it after forming the neurospheres or
So
prior to forming?
while making the neurospheres itself, that we add tamoxifen during the process of making neurospheres. And we have done the experiments either ways, both during the formation of neurospheres and after the formation of neurospheres, that we have seen that with TRF2 knockout, this neurospheres are not formed.
But if that was the case, you would see that the neurospheres would stop growing, but yours seem to be completely, they've sort of disassembled.
Yeah, that's because they take this neuronal differentiation form and then they attach to the bottom of the flask and then they take this neuronal morphology.
I see. And also just wondering, have you looked at the mouse brain? Like what is the effect on the cortical
Yeah,
layers?
that's where we are little stuck at. We have not been able to do in vivo experiments with mouse brain. That is our plan next.
Okay. And do you see a complete, sorry, one last thing. Do you see a complete loss of your TRF2 in the mice? Because there was one slide where you were showing Nestin Cree and it looked like there was still TRF2. So I was just wondering, how complete
There
is
is
your loss of
some
TRF2?
sort of mosaicism. There is no complete loss.
Okay.
Yeah.
Sandhya, go ahead.
Does overexpression of rest bypass the requirement of TRF2? Can it?
Does overexpression of rest? Ideally, it should not.
But has that been tested? Because rest itself could have effects. I mean, I'm just thinking of TRF2 as something which sort of docks onto G quadruplexes, but obviously the effects are
there are certain genes where
in a specific location, right? They are not acting on all G quadruplexes. They are not acting on all genes. So I sort of wondered that are there effects of rest which depend on other proteins?
Yeah,
There
are certain genes where
in
rest
which case you would uncover that if you overexpress rest in the absence of TRF2.
That experiment we have not done, but ideally it should not recover the loss of TRF2. But I do believe that there are certain genes.
More than lack of TRF2, but more that can it turn on some of the neuronal genes that you're seeing independent of TRF2 if you have sufficient amounts of rest?
It should not. So TRF2 is essential for it to rest to be recruited to the promoters. So I think rest overexpression should not completely recover.
So all effects of rest go only through TRF2 and there are no effects of rest in neuronal differentiation independent of TRF2. That's your thesis.
To answer this better, we are doing a ChIP-seq analysis where we are trying to understand the whole profile of where TRF2 and REST binds. So we do see that certain genes are not through TRF2, but majority of those genes are through TRF2. That is what we see.
Nice. Okay, thanks. I don't have any other questions.
Okay.
Sounds good. Thank you. Deepa, Sandhya, any other questions from you?
No, all
No,
good.
okay.
Thanks.
Okay, we are well within time then. Wondering if there are any questions in the chat window. Yeah, lots of good words, impressive pace of work, great slides. Snehal has a question. Saujanya, if you want to quickly answer it online.
Yeah.
What are the number of cells considered to be a neurosphere?
So they form, we see them per 96 well, and we start with 5,000 to 7,000 cells per well.
I see.
Yeah.
And then when they grow, what do you consider neurosphere? Is it just macroscopic appearance?
First, macroscopic appearance, and also we quantify through IF whether it expresses neural stem cell marker and flow cytometry.
Okay.
Right.
All right. Okay, Snehal, maybe if you need more clarification, the chat window is always there. We encourage you all to keep it active. We still don't have a winner for our giveaway, so that's waiting to be grabbed. Okay, thank you so much, Saujanya. We'll move on to our next speaker, who is Shreyas. Shreyas, nice to see you. Your video is on. I can see you're putting your audio on. You can share
I'm sharing
slides.
my slides.
Perfect. We can see them. And go ahead, Shreyas. All the very best. So we have 20 minutes, Rutuja. Just keep a track. 20 minutes for talk and 10 minutes for Q&A. Sounds good.
So good afternoon all. I am Shreyas from Dr. Sunil Lakshman's lab at INSTEM, Bangalore. And today I will discuss my thesis work where I'm trying to understand role of a protein phosphatase in regulating adaptation to glucose depletion. So as we all know that cells growing in natural environment encounter fluctuating nutrient conditions. And to adapt to these changing conditions, cells rewire their metabolism. For example, rapidly growing cells such as cancer cells or free-living cells such as yeast or fungi preferably use glucose as a carbon source, and these cells utilize glucose by the process of glycolysis and they ferment it to ethanol or lactate. But if these cells experience glucose depletion, these cells rewire their metabolism and they start utilizing other alternative carbon sources. These alternative carbon sources are utilized by processes such as gluconeogenesis and mitochondrial respiration. So this process of metabolic rewiring allows these cells to grow even in the absence of glucose. So this metabolic switching helps in adaptation. And this switching is very tightly regulated by signaling regulators. And in the context of nutrient fluctuation or glucose depletion, regulation by reversible protein phosphorylation is very well studied. So reversible protein phosphorylation is carried out by opposing activities of protein kinases and phosphatases. And kinases, which are the writers of this modification, are very well known to respond to changes in nutrient availability and to regulate metabolic outputs. And kinases such as TOR kinase, AMPK kinase, and protein kinase A are very well studied for their role in regulating metabolism in response to nutrient availability. However, protein phosphatases are poorly studied and underappreciated, and their role in regulating metabolism in response to nutrient availability is limited to only a few examples. So in my PhD, I was interested in identifying protein phosphatases that enable adaptation to glucose depletion. And to address this question, we used yeast Saccharomyces cerevisiae as a model system. And here you can see the growth curve of yeast Saccharomyces cerevisiae. As I mentioned, yeast cells preferably use glucose as a carbon source. So when sufficient glucose is available, these cells utilize glucose by the process of glycolysis and ferment it to ethanol. And during this phase, these cells grow exponentially. But if these cells experience glucose depletion, these cells undergo... I'm in a meeting, sorry. I'm in a meeting. Sorry for the interruption. So as I was saying that cells preferably use glucose as a carbon source. And during this phase, these cells grow exponentially. But if these cells experience glucose depletion, they undergo a metabolic adaptation known as dioxic shift. And during this dioxic shift, these cells rewire their metabolism. And in this post-dioxic phase, cells utilize other carbon sources such as ethanol and amino acids through the process of gluconeogenesis. And in this phase, cells also make storage carbohydrates such as trehalose and glycogen, which help them survive in these glucose deplete conditions. So I was interested in identifying protein phosphatases that enable this metabolic adaptation. And to identify protein phosphatases that regulate adaptation to glucose depletion, we carried out a targeted screen. So for the screen, we generated a deletion library of protein phosphatases in yeast. So for this, we generated individual knockout mutants of non-essential phosphatases, and we screened these mutants. So for the screening, we culture these mutants in high glucose conditions. We let them grow for 24 hours, and by this time point, the glucose is already depleted and the cells are in post-dioxic phase. And then from these mutants, we measure trehalose accumulation as a readout of post-dioxic metabolism. As I mentioned that trehalose accumulation specifically occurs in glucose deplete phase, and hence we can use trehalose as a readout of post-dioxic metabolism. So we carried out the screen, and here I'm showing the relative trehalose levels in the mutants compared to wild type cells. So from our screen, we identified that deletion of few phosphatases showed a reduction in trehalose accumulation, while deletion of few phosphatases led to an increase in trehalose accumulation, while most of the phosphatases did not show any change in trehalose accumulation. So from our screen, a notable hit was this particular phosphatase PPG1, whose deletion showed a maximum increase in trehalose accumulation. So this PPG1 phosphatase is known to regulate mitophagy and TOR-C2 signaling. However, its role in regulating metabolism is completely unknown. So we decided to study its role in regulating metabolism and adaptation to glucose depletion. So as I mentioned that this particular phosphatase regulates trehalose accumulation in the post-dioxic phase, we asked how the carbon allocation to other metabolic arm changes. So for this, we carried out metabolomics. So specifically, we culture wild type and PPG1 deletion cells in high glucose medium. We let them grow till the post-dioxic phase, and then we carried out LC-MSMS analysis. So from our MSMS analysis, we found that in the PPG1 deletion cells, the end products and intermediates of gluconeogenesis were increased. Also, other metabolites that are derived from this gluconeogenesis end products were also increased. Like for example, there was an increase in UDP glucanac, which is a precursor of chitin, and it is a output of gluconeogenesis. Also, we observed increase in UDP glucose, which acts as a precursor for trehalose, glycogen, and cell wall. So overall, our metabolomics data suggests that in PPG1 deletion cells, there is increased gluconeogenic flux and increased carbon allocation towards gluconeogenic outputs. So to unambiguously confirm this data, we carried out a metabolic flux experiment using a pulse label of 13C acetate. So again, for this flux experiment, we culture cells in high glucose, let them grow till the post-dioxic phase, and then we pulse the cells with 13C labeled acetate. So acetate is a 2-carbon compound, and here in this case, both the carbons of acetate are labeled with a 13C carbon, which is a heavier isotope of carbon. So once acetate enters the cell, it gets converted to acetyl-CoA, then it enters the TCA cycle, and then it enters gluconeogenesis. So we traced the incorporation of this 13C carbon from acetate into the intermediates of gluconeogenesis and other end products. And we found that the incorporation of 13C carbon in the end products of gluconeogenesis was increased in PPG1 deletion cells. Also, we observed increased incorporation of 13C carbon in other outputs of gluconeogenesis in these PPG1 deletion cells. So this data confirms that in the PPG1 deletion cells, there is increased flux towards gluconeogenesis and increased carbon allocation towards this gluconeogenic outputs. So to summarize so far, using a targeted screen, we identified a protein phosphatase PPG1 as a regulator of gluconeogenic carbon flux and a regulator of carbon allocation in the post-dioxic phase. So next, we wanted to identify what is the molecular mechanism through which this phosphatase regulates gluconeogenesis. So protein phosphatases are usually promiscuous and they lack the specificity towards their substrates. And they usually partner with regulatory proteins which direct them towards their substrates and helps them dephosphorylate the targets. So we thought identifying the interacting partners of this phosphatase will help us in understanding the mechanism of regulation. So for this, we tagged this PPG1 phosphatase with a flag epitope and carried out flag affinity immunopurification. And using proteomics, we identified the interacting partners of PPG1 phosphatase. And from our proteomic screen, the top hit we got was this particular protein FAR11. So this protein FAR11 is a component of a multi-protein complex known as FAR complex. So this complex is made up of 6 proteins which are non-covalently held together, and it localizes to outer membranes of ER and mitochondria, and it faces the cytosol and functions as a cytosol facing signaling hub. So we identified the interaction between PPG1 and FAR complex, and couple of previous studies have also identified this interaction in other growth conditions. So our experiment corroborates those papers and this data confirming the interaction between PPG1 and FAR complex. And those papers had hinted towards the possibility that this phosphatase is involved in the assembly of this complex. So we decided to study this in detail. So as I mentioned that this complex is held together by non-covalent interactions, and there is a possible role of this phosphatase in its assembly. So we tested this. So for this, we immunopurified FAR8, which is a component of the complex, and we checked for co-immunoprecipitation of FAR11, which is another component of the complex. So as you can see here that in wild type cells, FAR11 co-immunoprecipitates with FAR8. But when we study this interaction in a mutant of PPG1 that lacks the catalytic activity, we can see that the interaction between FAR8 and FAR11 is drastically reduced. So this suggests that PPG1 catalytic activity is required for interaction between 2 components of the FAR complex and overall assembly of the complex. So since PPG1 is a phosphatase, we ask the question, does PPG1 dephosphorylates components of FAR complex to help for its complete assembly? So we address this. So first, we used a crude readout of for checking the modifications on components of FAR complex. So we just looked at their electrophoretic mobility on a SDS gel, and we observed that specifically for this protein FAR11, when we study its mobility, we observed that in the mutant of PPG1, the mobility of FAR11 is reduced. So this suggests that FAR11 gets modified in a PPG1 dependent manner. So we wanted to identify these modifications and the specific sites at which FAR11 gets modified. So for this, we immunopurified FAR11 from wild type and mutant cells in large sufficient quantities, and we carried out proteomics analysis. And we found that in the PPG1 mutant, these FAR11 phosphosites were specifically increased, suggesting that PPG1 is involved in dephosphorylation of these sites. That's why in the mutant of PPG1, the phosphorylation of these sites is increased. So this confirms that PPG1 dephosphorylates FAR11, and this particular dephosphorylation is required for interaction between FAR8 and FAR11 and the complete assembly of the FAR complex. So to summarize so far, we identified PPG1 as a regulator of gluconeogenesis, and we also showed that PPG1 is required for the assembly of FAR complex. So now we have identified interaction between PPG1 and FAR. We ask, is FAR complex also required for this regulation? So for this, we generated deletion mutants of components of FAR complex, and we measured the amounts of these metabolites in the post-dioxic phase. And similar to PPG1 deletion cells, the amounts of these metabolites increased in the deletion mutants of FAR complex. So this suggests that deletion of FAR complex completely phenocopies deletion of PPG1. So our data suggests that PPG1 mediated assembly of the FAR complex is required for regulating the gluconeogenesis and allocation of carbon to gluconeogenic outputs. So since now we show that PPG1 regulates gluconeogenic carbon flux, we also asked, is PPG1 and FAR required for regulation of metabolism in glucose replete phase when sufficient glucose is available? So for that, we studied glycolytic metabolism in these deletion cells, but we did not observe any change in the metabolism. So this suggests that this regulation specifically happens after glucose depletion. So we think that maybe glucose signals to PPG1 and or FAR complex and thereby regulates their activity to regulate gluconeogenesis. So for this, first we looked at the amounts of PPG1 and its activity. We found that the amounts and activity of PPG1 phosphatase do not change in both low glucose and high glucose conditions. But when we looked at the amounts of FAR complex, we observed that in high glucose conditions, at the 6 hours time point, when sufficient glucose is available, the amounts of FAR complex are low. But after 24 hours of growth, when the cells are in post-dioxic phase, the amounts of FAR complex increase. So this suggests that amounts of FAR complex increase as glucose depletes. Also, when we add glucose to these post-dioxic phase cells, the amounts of FAR complex again decrease. So this suggests that glucose availability regulates the amount of FAR complex. So to summarize so far, this PPG1 FAR assembly, which is localized at ER and mitochondria, is kept in low amounts when sufficient glucose is available, and it does not interfere with glycolytic metabolism. But when glucose depletes, the amounts of FAR complex increase, and it is involved in regulation of gluconeogenesis. So we have identified a new signaling axis where a protein phosphatase and a scaffolding complex respond to glucose availability to regulate metabolism. So next we ask, is this signaling mechanism that we have found, is it actually required for adaptation to glucose depletion? So to address this, we designed a competition experiment. So for this, we mixed wild type and mutant cells in equal proportion, and then we challenged them with changing glucose conditions. So specifically what we do, we let them grow in high glucose conditions for 24 hours. So by this time, the glucose completely depletes, and then we shift them to a glucose-rich medium. And again, we let them grow for 24 hours, and we again transfer them to glucose-rich medium. So we repeat this cycles of transfer. And after each transfer, we measure relative amounts of wild type and mutant cells and calculate the fractional composition. So what we observed that during the course of our competition experiment, the wild type cells completely outcompeted the PPG1 deletion cells. So this confirms that the PPG1 FAR signaling mechanism that we have found is required for adaptation to changing glucose conditions. So to summarize what we have found, so we find that irrespective of the glucose availability, PPG1 phosphatase dephosphorylates FAR11, and this dephosphorylation is required for its interaction with this FAR8 and total assembly of FAR complex. In high glucose conditions, the FAR complex is kept in low numbers, but as glucose depletes, the amounts of FAR complex increase, and PPG1 FAR together regulate gluconeogenic carbon flux and outputs of gluconeogenesis. And this regulation of carbon flux is required for efficient adaptation to glucose depletion. So in the future, we are interested in understanding the molecular players that mediate this regulation. So with this, I'd like to stop and thank my thesis supervisor, Dr. Sunil Lakshman, my thesis committee members, my lab members, Lavanya, who helped me with this project, all the funding agencies, INSTEM and CBS, Manipal, and India Bioscience for the opportunity to present. Thank you.
Very
I guess
nice,
I'm
Shreyas.
within the time.
Some
Yeah, so I can take questions.
classical screen, and then your assays. Over to you, Deepa and Sandhya.
Yeah, very nice talk, Shreyas. I was just wondering, you showed the competitive, sort of the competitive growth assay in the end. But individually, does loss of PPG1 actually alter growth? Is the growth rate reduced, and is that very dependent upon the type of media it is in?
Yeah, so what we observe that when we culture these PPG1 deletion cells, till the glucose is available, they grow exactly similar to wild type cells. But as glucose depletes and they enter the post-dioxic phase, we observe a very specific growth defect. So this suggests that this regulation is very specific to glucose deplete conditions, and it is actually required for growth in that phase.
So also, just wondering, PPG1, because it's a phosphatase, and this switch between, even in 2 hours of addition of glucose in the depleted media, you seem to be able to see a reduction in these levels, right? I was wondering, are there any known post-translational modifications of PPG1, and how is that influenced by the presence of glucose?
Yeah, so we have looked at the activity of PPG1, and we find that the activity and the levels of PPG1 do not change even though the glucose level changes. But what we have observed that the amounts of FAR complex change, and also we have identified, we have observed that FAR complex gets post-translationally modified as glucose depletes. And these modifications happen on this protein FAR11. So this we have observed through Western blot just by looking at the mobility shift on the protein, but we do not know the identity of these modifications and what these modifications are. Yeah, but clearly there is a response to glucose depletion and some upstream regulation is happening.
And have you seen any changes in mitochondrial morphology?
So we have looked at the mitochondrial activity. We don't see any change, but we haven't looked at the morphology of mitochondria. We have used like bulk readouts like MitoTracker Red for that, the intensity of the membrane potential, but we do not see any changes in that.
Okay, thanks.
Thanks.
Over to you, Sandhya.
Yeah, I also had one very similar questions. Then have you looked at the ER at all? And because that's one of the things that you were saying, that this is both at the ER interface, and have you looked at ER mitochondrial contacts? And I'm very intrigued by the involvement of the ER in this process.
So what we have done is that, so there are two subpopulations of FAR complex, one at ER and one at mitochondria. So we have CRISPR engineered E strains, and such that in one strain it will be only localized to ER. It will be targeted only to ER, and in other strain it will only be targeted to mitochondria. But what we see that both these strains behave like wild type cells. So even though like the localization of FAR complex to ER or mitochondria is not important for this regulation. And just so we are thinking that
And how
maybe
did you attach it to the ER?
So FAR complex, one second, I'll just go back to the model of FAR complex. So these two proteins of the FAR complex, FAR9 and FAR10, they have a transmembrane segment that gets inserted into the membrane of ER and mitochondria. So what we did, we replaced this transmembrane segment with that of a protein that only goes to mitochondrial outer membrane. So that way we were able to direct it to mitochondria and ER separately. But we observed that it's targeting to ER or mitochondria doesn't make any difference to this regulation. So we think that maybe the downstream substrates or targets are localized in the cytoplasm and accessible by both the subpopulations of FAR complex present at ER and mitochondria.
But you do need it. So if you have cytoplasmic FAR 9 or 10, that won't do the job.
Yeah, so we did that experiment also. So we detethered the FAR complex. We deleted the transmembrane segments of both these proteins. So in that case, the cells behave like PPG1 deletion cells. But we also observed that the interaction between these FAR complex components was lost. So maybe we think that this attachment to the membrane acts as a nucleation point where these other proteins can come and dock on top of each other. And this interaction doesn't happen in the cytosol that well, because it's all diffused.
Have you sort of tested that hypothesis by targeting these proteins to any membrane, like the plasma membrane, have it face on the inner surface, and then you can get this nucleation happen?
No, that is one of the plan, but we haven't done that experiment yet. Yeah.
Karishma, you are muted.
Yeah, thank you. Thanks for letting me know. All right, I said thank you, Sandhya, Deepa, and Shreyas. I said great talk, Shreyas, and fielded the questions also very nicely. We do have a little time. You finished well within time. So Snehal has some interesting questions in the chat window. Do you want to take them? It's a good one. First one is, what was the high glucose that you used? Like, what was it?
So it is
Molar
2%,
concentration.
110 millimolar.
That was what you called high glucose.
Yeah.
Okay, you can respond further to Snehal also in the chat window.
Yeah.
And there's another one, Shreyas. Have you tracked anything on the lines of mitophagy with respect to PPG1 FAR complex?
So PPG1 FAR complex is involved in regulating mitophagy, but in yeast, mitophagy happens in severe nutrient deplete conditions, like such as severe nitrogen starvation and glucose starvation. And the conditions, like our experimental conditions, do not have such severe starvation. In our experimental conditions, we still have sufficient amino acids in the medium, so we don't expect any mitophagy to happen at this, like in these conditions. So, and also we have ruled out the component of mitophagy by deleting some of the components of regulators of mitophagy, such as ATG32. And we show that this regulation is independent of FAR complex's role in regulating mitophagy.
All right. Yeah. Any other questions? I'm not sure. Snehal says thanks to you. Yes. Okay, you can maybe, if there are more questions, take them in the chat window.
Sure.
But we are
Thank you so
well
much for
within
the
time, so
opportunity.
thank you. Thank you. All right. Our next speaker is Sanjana. Sanjana also has a very interesting title. She calls it ARID2's yin yang effect. So tell us more about that, Sanjana. It sounds very interesting. We can see your slides. You might want to make it slideshow.
Yeah.
And yeah,
Can you hear
sounds
me?
good. Yeah, we can.
Yeah.
We can.
Also,
You can.
Sanjana, hello. Also, Sanjana, please use pointers.
Yeah, sure.
Sure. And Sanjana, do you want to put your video on?
No, actually, my video is not working somehow.
No worries. Go ahead. No problem. Sounds good. All the best.
Okay, good afternoon, everyone. So before starting, thank you to India Biosciences for giving me this opportunity. So as introduced, so I will be talking about ARID2's yin-yang effect. I am from Center for DNA Fingerprinting Diagnostics, and I work under the supervision of Dr. Muralidharan Bashya. So before beginning, so we all know inside cell DNA is compactly wrapped around histones and other proteins. So for gene expression, it has to open. So this opening of uh, tightly packed DNA is done by certain chromatin remodeling complexes. One such chromatin remodeling complex is YSNF chromatin remodeling complex that utilizes ATP hydrolysis to slide or evict nucleosome so that it is further available for the transcription factors to do gene expression. So this YSNF chromatin remodeling complex is of three types, majorly BAF, PBAF, and NCBAF. So these three complexes have certain subunits which are core and ATPase module in common, like BAF 150, BAF 120, BRG1, while certain components like ARID1A, 1B, ARID2, then BRD9, these are specific to, like for in case of ARID1AB, it is specific to BAF, while for PBAF, ARID2, etc. So today's story will be regarding ARID2. So it is a AT-rich interactive domain protein 2, and it specifically provides stability and formation of PBAF and drives this entire complex to the DNA for this processivity. So since past 20 years, when the sequencing studies had increased, it has shown that multiple mutations in SWI/CNF subunit causes human cancers. So if you focus in this specific heat map here, we can see that ARID1A, ARID1B, PBRM1, then ARID2, these all are highly mutated in lung cancer, colorectal cancer, breast cancer, etc. So, from our study, we had shown that while performing exome sequencing studies in EOCRC sample type, which is early onset colorectal cancer type, and which is highly predominant in India, that 11% of samples showed ARID2 inactivating mutations. So, these were the mutations which we reported. Further, in vitro and in vivo experiments on ARID2, we showed that when we knock down ARID2, it shows higher growth. Thus, it is a driver of cancer. So, if ARID2 is not there, colorectal cancer was happening, suggesting that ARID2 is a classical tumor suppressor gene. Interestingly, when we were performing certain rescue experiments in knockdown and presence of wild type ARID2 background, one mutation, which is this 989 frame shift mutation, which was leading the protein to truncate around 1010 amino acid. So this specifically showed that it was highly migrating in transwell migration assay. So as I told that ARID2 is a classical tumor suppressor, but interestingly, this mutant was behaving otherwise. So the next question was what this mutant is doing. So as I told that it is a truncating mutation, so the first thing we checked was from the other classical cancer data sets whether this type of truncating mutations occur or not. So interestingly, in the entire 1835 amino acid long protein in this specific region where our cohort mutation is lying, there are multiple truncating mutations. Approximately 34% of truncating mutations lie in different cancer types and different pan cancer data sets, like we took from TCGA GDC portal and CBio portal data sets. So interestingly, also we have mined data from DepMap portal where we find cancer cell line data sets and also multiple cancer cell lines like neuroblastoma cell lines, colorectal cancer cell line, lung cancer cell line. So of 29 cancer cell lines showing truncating mutations of ERIC2, 11 lie in this specific region. So interestingly, it is known that from literature, the N-terminal domain, which is the ERID and RFX domain, in this specific domain, it is known to bind the other PBAF complex components. So what happens is first the core module binds, then the ERID2 binds, and then this specific domain facilitates binding of the other components, and thus the PBAF complex is formed completely. So the next question was, this region is intact even though in the truncated protein, so it might form the residual complex, or it is doing something to the nuclear complex and thus affecting the tumor growth. But when we performed immunoblotting after pull down experiments, we could see that the wild type protein was able to pull all the core component ATPase module and the PBAF specific module, while the 989 is unable to do so. So it is not pulling any of the core component or any nuclear PBAF complex components. So interestingly, when we further checked its localization, so the wild type protein is located in nucleus as it does the chromatin remodeling functioning, while this 989 frameshift mutant was happily sitting in cytoplasm. So this excited us, and we further thought that maybe it is in cytoplasm and it is bringing the complex to cytoplasm, hampering its nuclear function. But again, if you focus here, when we did sequestration assay by checking for all the multiple components in IF, we can see that BRG1, BAF155, BRD7, everything is in nucleus, while the ERID2 989 is in cytoplasm. But when in case we are overexpressing the wild type ERID2, it is in nucleus and other components are also in nucleus. So it has the mutant protein has nothing to do with its nuclear functioning and the PBAF complex formation. So interestingly, the next question is, because this is a half the size of the wild type protein, where is the NLS? Because this is happily sitting in cytoplasm. So we opted for in silico mapping by cNLS mapping for bipartite NLS sequence. So why we selected bipartite NLS is because ERID1A, 1B, which is a homologue of ERID2 and is present in the 1488, which is not having both the constructs. One is 1538. This is just having the 1488 construct, and the 1625, which is in between the two. So interestingly, if you see here that the 1488, which is not having both the NLS, was sitting happily in cytoplasm, while when we are just incorporating the 1488 to 1520 stretch, it is going in nucleus, suggesting that 1488, which is NLS1, is possibly the canonical NLS of ERID2. So the next interesting thing was, as we checked previously in the rescue experiments, the oncogenic growth. So we opted for more tumorigenic assays by MTT growth and also wound healing. And now we did with all the construct, we have the wild type ERID2 construct and also the two cytoplasmic variant of ERID2, which is 989 and 1488. So in both the background, so we generated ERID2 knockout HCT11 cell, and in both the background we can see that 989 and 1488 show higher fold increase in cell growth. So the next question was, what is the region which is responsible for gain of function activity? So we chopped the protein more. So one protein we selected was 636, another one was still 725. So these were again selected on the basis of frequent multiple mutations in cancer cell lines and also from C bioportal tumor data sets. And this was highly present 725 truncations in skin cancer as well as endometrial cancer type, where ERID2 is highly mutated. And here also, if you now see, interestingly, this 989 protein is showing tumorigenic effect, while 636 and 725 truncated versions of ERID2 are unable to show any growth or wound healing, anything. So this suggests that there is a difference between this 989 cytoplasmic variant and this. So the main difference between these is the GLN domain. So next, the main question was whether this is a generalized phenomena in cancer or colorectal cancer specific phenomena. So what we decided to do all the construct
Sanjana, we cannot hear you.
Yes, it's possible that she's lost network at her end. Seems to be like that. We'll just give it a few seconds. In the meantime, Sanjana, we are waiting for you to come back. In the meantime, it's a good time to say that we need more questions in the chat window because that giveaway has to still happen at the end of the session. So keep it active, keep your questions flowing in. I think now Sanjana has dropped. No, she's there. Sanjana, can you hear us?
I don't see her in the list. I think she's not yet joined back.
And there is one Sanjana Sarkar though. Yeah. We'll just probably give it a few seconds.
And now I can see Sanjana Sarkar. Sanjana, are you there? Can you hear us?
We just wait a little bit. It's probably an internet or a power cut issue. Yeah, I think she's coming back in. Rutuja, you just want to keep a check on the waiting room? She's coming back and I just let her in. Yeah. Sanjana, are you back in?
Hello?
Sanjana, I think you've joined from 2 accounts.
Hello, now can you hear me?
Yes, we can.
Yeah,
Perfect.
we are able to.
Just share
Sorry,
your slides again.
Yeah, just one second. Yeah, can you see?
Oh, yes, we can. Go ahead.
Yes. Yeah, wait, just. So, so where was I last when it went away here?
The next slide, actually.
Okay.
Hmm.
Yeah. So as I was whether the mutant ERI2 is causing this specific oncogenic phenomena in colorectal cancer or it is a generalized phenomena. So we opted to do all the experiments in lung cancer again because ERI2 is known to be a tumor suppressor in lung cancer. So we opted to do all the experiments in H1299 lung adenocarcinoma cell line, and we did with all the truncated mutations which we had from 636 to wild type. And interestingly, we could see that again here like 989 and 1488 are causing the gain of oncogenic function while 636 and 725 are unable to do so. So as in the previous slide I was stating that the difference between these and this is the GLN domain. So the GLN domain is a glutamine proline rich domain. So it has high number of glutamine proline and it is known to form a intrinsically disordered region for a like a novel interactome group and forms a liquid-liquid phase separation. So the next question, as I'm stating that, what is the mechanism by which this mutant ERI2 is functioning? So we opted for tandem affinity purification followed by mass spectrometry. And these are certain list of proteins which were highlighted and the red ones marked are known to be highly important for tumor formation. So one such protein which we interested was ACAP8L, which is a kinase protein anchoring 8-like protein. So this ACAP8L was recently known to be activating mTOR pathway. Also, ACAP8L has a ACAP95 domain, which also contains high glutamine and proline rich domain, so it can interact with any kind of ideal target. And as well, this ACAP8L is known to have direct target of cyclin D and cyclin E1. So interestingly, if you see here, this ACAP8L only interacts with 989 ERIT2, while the wild type and empty vector is unable to do so. And in the reverse pull down as well, when we are pulling down ACAP8L, 989 ERIT2 is pulled down. So 989 is a direct interactor of ACAP8L. So interestingly, when we perform TMA followed by IHC experiments, 13% of samples showed cytoplasmic localization of ERIT2. So as I have been telling that ERIT2, we from our lab and also from previous literature, it was shown that it is a tumor suppressor. So obviously 18% samples also showed inactivation of ERIT2. So as told that when we generated ERIT2 knockouts, so we could see that yes, when ERIT2 is not there in colorectal cancer, they are showing high growth. So what is happening is when ERIT2 is mislocalized to cytoplasm, it is showing higher growth, and when ERIT2 is not there in colorectal cancer tumor samples, it is also showing, because of inactivation, higher growth. So wild type is responsible for tumor suppressive activity, while the mutants are responsible for tumor formation. So now in case of inactivation, like what is happening to the complex? So we saw that in case of the cytoplasmic variant, it is entirely in cytoplasm and it has nothing to do with the complex. So when we checked further that when wild type ERIT2 is knocked out, what is happening to the complex component? We could see that the PBAF complex component, which is BRD7, it is going down, while the CBAF complex component and the core module are intact, suggesting that when ERIT2 is knocked down or knocked out, the PBAF component complexes are going down. Why? And also, interestingly, when we did fractionation experiments, we could see that not only it is getting degraded, but it is mislocalized to cytoplasm again. So what is happening here is when ERIT2 is not there, nothing is going wrong with the other components like the NCBAF or the CBAF component, while the PBAF complex component is degraded, and whatever protein is left for BRD7 or PBRM1 or PHF10, which are specific subunits of PBAF complex, are either degraded or mislocalized to cytoplasm. They are not present at all in nucleus. The same we have observed in IF as well. So here I am probing for two specific PBAF complex component proteins. Here we can see that PHF10 and BRD7 in 8CT116 ERIT2 knockout, they are mislocalized to cytoplasm and they are much more lesser with respect to the wild type ERIT2 8CT116 cell line. But if you see one ATPase module, there has nothing to do in case of 8CT116 ERIT2 knockout. So they are happily sitting in nucleus. So what is the story so far going on is that when ERIT2 is present, it forms the PBAF complex and it is helping in doing the chromatin remodeling. When ERIT2 is not there, so the residual PBAF complex is degraded or it is not formed or mislocalized to cytoplasm. And interestingly, when ERIT2 has mutation like the truncating mutation which we found in our cohort, or from the TAPMAP portal and CBio portal data, when we have the truncated protein, it is mislocalized to cytoplasm and thus activates the mTORC pathway by the ACAP8L axis and thus forms the tumor. So this is the yin-yang effect of ERIT2. So when it is in nucleus, it happily forms the complex and thus drives tumor suppression. But when mislocalized to cytoplasm and has mutations, it is driving oncogenesis. So this is the final slide. So I want to thank my supervisor for giving me opportunity, DAC members for giving the critical comments, and these are the ones from which I got to know about the mass spec and the constructs. So Mr. Vishwakalyan, the IHC experiments, and obviously my lab members for always keeping a happy environment and helping anytime I need. Thank you.
Nicely done. Yeah, sure. Go ahead, Deepa.
I was just wondering, the knockout versus the mutant forms of your ARID2
Yeah.
seem to be doing very different things with respect to regulating the localization of the PBAF components. So when you have a knockout, the PBAF complex seems to be, or the whatever other proteins are part of the complex, they seem to be present in the cytoplasm, as your immunocytochemistry also shows. Whereas earlier you'd shown this slide of looking at other components in the presence of the mutants, and they still seem to be nuclear localized. So just wondering, when the mutation is there, is it doing different things? Is it sequestering proteins in a different way, which then prevents their natural function? And have you looked at sort of nucleosomes and occupancy, etc.? So because that would be your direct readout of the function of this complex, right? So how does that differ between the knockout versus any of the mutations you're looking at?
Yeah. So actually, ma'am, so when we are knocking out ARID2, so in this case, what is happening? So if you see in wild type also, little bit is present in cytoplasm, correct? So what I'm stating is when in knockout ARID2 is not there, PBAF complex is not at all formed. But while in case of the mutant ARID2, it is completely different from the PBAF complex or chromatin remodeling. So it is happily sitting in cytoplasm and it is activating other pathways or interacting with other protein which has nothing to do with the wild type ARID2. And also, like in case of knockout, currently we are performing the like cut and run and ataxic experiments, which will give us the direct link of what is happening to the accessibility. But like from all the experiments and whatever, like currently I'm performing QRT PCR for targets and all. So from that, what I can say that the wild type ARID2 and the mutant ARID2 completely behaves differently.
Okay. And I was wondering, so these mutations were originally identified in colorectal cancer. Is that right?
Yeah. So the one which I have
the frame
showed.
shift and
Yeah,
deletion,
correct.
right?
But
Yeah.
the like we have checked from multiple data sets, and like there are truncating mutations like in pan cancer data sets as well.
So, so I know you looked at some gene expression changes, right? Of some for some genes. That's where you identified the ACAP. That was gene expression, right? Did I get mixed up? We lost.
Sanjana, did you get that question?
Hello.
Yes, Sanjana,
Sanjana, are
did
you
you?
there?
Yeah, can you repeat once again?
Yeah. So I was wondering if you looked at, sort of global gene expression changes to see how that would be affected in the presence of these mutations.
Yeah, so for that, what we are currently doing is like we have generated in knockout background stable cell lines. So in that we are performing RNA-seq, and also like I was stating that the cyclines and like there were cyclin E, cyclin D1. These are like greatly enhanced in case of the 989 one, but not in case of wild type expressing one.
But are the mutations present in a homozygous fashion, or is it heterozygous in the patients?
Yeah, so this is again like previous talks. I will state that this is also like a dominant negative. So in the tumor sample in which we found this was one allele was completely lost, and the other one was having this truncated mutation.
Okay.
How do you conclude that it's dominant negative based on that?
Yeah, so with respect to like in presence of wild type ARID2 as well, when we are expressing 989, it
was
overpowers the
lost.
wild type.
You said
Hello?
one allele was lost and the other allele was mutated.
Yes, correct. Yeah.
Go ahead, Deepa.
No,
Sorry.
I'm good. I'm all done.
Okay, so you said about 34% of your colorectal cancers had some mutation in ARID2. I was wondering, have you looked at, this is again going back to something Deepa asked you already about how mutants seem to behave somewhat differently from wild type. And one of your data suggested that your wild type might be important for early on in just getting the cells. Is that, I hope I understood that correctly. If not, please correct me. Early on, your ARID2 was important for making sure that you had cell division, but later on, the mutant one sort of tipped it over for basically uncontrolled growth. Did I understand that correctly?
Just 1 second, ma'am. Can you repeat the question one more time? I cannot. Hello?
So I said that your ARID2 initially you seem to require ARID2 for maintaining some sort of cell division, right? Wild type ARID2.
Not cell division, but like it helps in chromatin remodeling. So,
Yeah.
like it,
And it
yeah,
was only later on that the
yeah,
mutant, so you needed ARID to function to sort of help maintain your cells in some sort of healthy state.
yeah, correct. Yeah, so the wild type ARID2 maintains the stability and the chromatin remodeling, but like in presence of wild type ARID2 also, if you're putting mutant form or the wild type ARID2 is knocked out, so now either it is not able to form the chromatin accessibility, thus the gene expression is not happening, or this mutant ARID2 is driving tumorigenesis by activating different signaling pathways.
So that's where your gain of function hypothesis comes from.
Yes.
Okay. Okay. So have you seen colorectal cancers with upregulation of ARID2? So the question I'm asking is, is overexpression of ARID2 alone sufficient to sort of tip the balance over to cancer, or do you need to have these mutant versions?
Yeah, so for answering this, so when we check for specifically the wild type ARID2's expression profile, we don't see high expression, obviously because it's a tumor suppressor. But when I checked for these, the mass spec proteins and all, so I
No
could see,
expression, any change
yeah,
in expression.
yeah, so correct. So the mutant ones or the truncated ones show high expression in colorectal sample sets.
No, but what about the non-34% version that you have, right? Not the ones which have the mutations in ARID2, but the ones which are not mutated. Do you also see changes in gene expression of ARID2?
No, in that I don't see.
I see.
Yeah, yeah.
You also talked about phase separation as potentially
Yeah.
being important. So where does that phase separation takes place? Does it take place where it's trying to remodel? Does it take place in the cytoplasm? I was not
Yes.
clear to frame it from your talk.
Yeah, so basically, because of the GLN domain, it basically is phase separating in cytoplasm. So it creates like, wait 1 second, I think I can share.
Why does it not phase separate in the nucleus? I don't understand that.
No, so from our experiments, what we have seen is in case of like when we are over expressing the wild type also in nucleus after like 48 hours or so, it can form the phase separated punctures, but that is because of basically because of the overexpression thing. But in case of cytoplasmic variant, so what is happening is in case of when we are deleting till 1488, the NLS is not there, so the GLN domain is much more accessible.
Oh, I see.
Yeah, so in that case, so in this slide,
when the protein is going through the nuclear pore, should it not be much more unfolded so the NLS is exposed and then it sort of enters?
Yeah, so yeah, ma'am. So you're correct. So when it enters, then again it needs a proper folding and just the end terminal accessible for the other components to bind. So in nucleus, it is very well folded and like very like completely structured. So which is not happening in cytoplasm.
That is your
Because
hypothesis to explain this difference.
yeah, correct. Yes, because it is.
You have to read and probably.
Okay, all right. Thank you. Thank you for the clarification.
Thank you so much, Sandhya, Deepa, and to our speaker as well. Thank you so much, Sanjana.
Thank
Sanjana,
you.
if there are any questions, you take them in the chat window. That was a very nice presentation. Lots of discussion that came out of it. So thank you very much. We'll now move on to our next speaker, which is Prajna Dipta, who's going to be talking about neuronal specific modifiers in C. elegans under heat stress. So, Prajnadipta, are you here?
Yes, yes. Am I audible?
Great. Yes, you're audible. Maybe you can
Yeah.
put your video on if you can. Yes, we can see your slides.
Yeah. You can see.
Yes, and we can see you too. So, we made it work. We made it all work. Audio, video, and slides. Excellent, Prajnadipta. All the best. Go ahead.
Thank you so much. A very good afternoon to all. I am Pragyandipta from IIT Mandi, going to present my work, and I work in the lab of Dr. Prasad Kasturi. So at the outset, I would like to thank the organizers, India Bioscience, for giving me an opportunity to present my work. And I hope so by the end of my presentation, I will get some valuable suggestions and feedbacks from the expert mentors. So today I will be presenting my work titled Unveiling Neuronal Specific Modifiers of Protein Aggregation in C. elegans under heat stress conditions. So I would like to start with a little background before going into the findings of my work. So the global population is aging rapidly, creating significant challenges for healthcare, economies, and societies. In the right side, you can see the WHO map shows the global distribution of people aged 60 and above and the increase of aged population from 2015 to 2050. Regions shaded in dark blue represents areas with a higher percentage of elderly populations, highlighting significant aging trends in Europe, North America, and parts of Asia and Oceania. Now the question is, what is aging and why we need to study it? So aging is characterized basically by the progressive deterioration of the body's physiological functions, which is associated with decreased health, increased incidence of degenerative diseases, and an increased risk of death. In order to study aging, various hallmarks have been discovered, where loss of proteostasis one of the major primary hallmark. Mostly, our lab works on the concept of proteostasis. So next, we will understand the concept of proteostasis. So maintaining proteostasis is essential for organismal health. Proteostasis is often termed as state of balanced functional proteome. Once a protein is synthesized, it has to be correctly folded in order to perform its physiological function. This folding is undertaken by a class of proteins called chaperones. Once the function of the protein is over, it has to be degraded. Proteostasis ensures that proteins are synthesized, correctly folded, they are targeted, and also degraded once their function is over. But many a times, cells perceive to various kinds of stresses, which results in the misfolding or unfolding of the proteins. As the misfolding or the unfolding of the proteins increases inside the cells, they clumps together and forms aggregates, which can cause proteotoxicity to the cells. When these events occurs in neurons, the consequences can be devastating. When there is proteostasis maintained inside the neuron is healthy. When there is aberrant proteostasis in neurons during aging or stress, it can lead to various neurodegenerative conditions like Alzheimer's, Parkinson's, Huntington's, amyotrophic lateral sclerosis, and further leads to a defective neuron. So now the question is, can we revive proteostasis to protect our neurons? So in order to assess a few of the mechanisms of proteostasis, we are using a model organism, which is a tiny worm called Caenorhabditis elegans, or in short, C. elegans. The figure shows here is a C. elegans, and it has various advantages as well as key features of using this model organism. So it's a free-living, small, transparent nematode. It has the capability of self-fertilizing, and it can be genetically manipulated. It has a very short life cycle, about 3 weeks, which is ideal for studying aging and age-related diseases. So it shares many genes and pathways with mammals. Lifespan mutants and disease models are also available. One of the key features is it grows at 20 degrees Celsius, which is the optimum temperature. When you talk about the sharing of pathways with mammals, there comes a conserved pathway, which is a insulin or insulin-like growth factor signaling, which is conserved from lower organisms to higher organisms. DAF2, which is the C. elegans homologue of the insulin-like receptor in mammals, gets activated by insulin-like peptides, which initiates the signaling cascade. Further, they inhibit the transcription factor of DAF16, which is a FOXO transcription factor, and HSF1, which is a heat shock factor, to enter into the nucleus. Under conditions of low insulin signaling, DAF2 is blocked, and DAF16 and HSF1, they are free and they translocates into the nucleus and activates its target genes. As I'm going to talk about the mechanisms of protein aggregation in further slides, I would also like to highlight other functions of DAP16 and HSF1 in terms of protein aggregation. The default pathway regulated by HSF1 identifies and breaks apart toxic aggregates. When HSF1 machinery is overloaded, however, a molecular pathway regulated by DAP16 comes into action, which results in the formation of less toxic aggregates. This we try to find out experimentally, which I will be discussing in further slides. With this background, I will put for the overall goal of the work. The overall goal was to identify protein candidates that play a role as modifiers of protein aggregation and quality control in neuronal tissues of C. elegans. And for my study, I am using a model aggregation prone protein, which is a modified firefly luciferase. In the neuronal tissues, there are two conditions I took. There are healthy and stress conditions. And the identified candidates, they might also play as modifiers of other protein aggregation and potential to modulate aging. So we are using a modified firefly luciferase as a model for protein aggregation. This model has been well characterized, which was shown by Gupta et al. In 2011, where two mutations are made in the N-terminal side of the luciferase protein to make it metastable. Further, it has been fused to GFP. As two mutations were made, we call it as a double mutant firefly luciferase, or in short, I'll be telling DM fluke. In order to study neuronal proteostasis specifically, we express the DM fluke in neuronal cells under the control of RGF1 promoter, which expresses DM fluke pan-neuronally. The figure shows that this figure shows it expresses in neurons. The beauty of the system is when the worms are subjected to heat stress around about 33 degrees Celsius for 1 hour, they form punctate-like structures. Those are aggregates of DM fluke. But when we put the worms back into the recovery temperature, that is 20 degrees Celsius, about 6 hours, the aggregates recovered, which allowed us to study mechanisms of neuronal protein aggregation quickly and efficiently. Then further, we recapitulated this in our lab, and we checked the fluorescence microscopy images with heat shock and the recovery. And we also observed the same as the previous, that in 33 degrees Celsius, DM fluke forms aggregates. And when we put the worms back into recovery, that is 20 degrees for 6 hours, the aggregates recover. Further, we confirmed with insoluble or soluble protein extraction. If there are formation of aggregates, it should come in the insoluble fraction. And if there are no aggregates, it should come in the soluble fraction. To check that, we used high-speed ultracentrifugation to separate insoluble and soluble fraction without heat stress and with heat stress. Here in the figure, T refers to the total protein, S is the supernatant, where there is soluble fraction, and P is the pellet fraction, where there is the insoluble fraction. We probe them with luciferase antibody. Without heat stress, we could see the band in the supernatant, but not in the pellet, suggesting that DM fluke is more in the soluble fraction when there is no heat stress. Further, we checked for the bands with heat stress, and we found that the band is present in the pellet fraction, suggesting that DM fluke is going towards the insoluble fraction when we provide heat stress. As I mentioned about HSF1 DAP16 previously, so next we wanted to check how HSF1 DAP16 regulates the DM fluke aggregation. In order to assess that, we generated a strain where we deleted HSF1 and DAP16 in the DM fluke background and checked for the presence of DM fluke aggregates using fluorescence microscopy. We did the imaging at normal temperature, a mild heat stress condition at 25 degree and heat stress at 33 degree for 1 hour, and recovery at 20 degree for 6 hours. At normal temperature at 20 degree, we observed that wild type and DAP16 mutants, there were no aggregates, but in the HSF1 mutant, there were few aggregates. When we checked in the 25 degree, we observed that wild type and DAP16, there were very few aggregates, but in the HSF1, there were increase in aggregates. When we give heat stress, as previously, we observed that in all the three strains, there are formation of aggregates. Further, we checked the recovery and found that wild type and DAP16 mutants, the aggregates recovered completely. But there were not at all recovery of aggregates in the HSF1 mutants. This shows that HSF1 is required for disaggregating the protein aggregates and also responsible for maintaining the conformational stability of DM fluke without stress conditions. Further, we wanted to check for the protein folding. For that, we performed specific luciferase activity, which typically refers to the luminescent signals produced by the luciferase enzyme normalized to a specific reference such as total protein concentration, which was obtained from the Western blot analysis. The luciferase assay, which I'm showing here, is at 20 degrees Celsius. At 20 degree itself, the activity for wild type is at 100%, followed by DAP16 mutant worms around 80%, and in the HSF1 mutant, it's around 60%, which suggests that in the HSF1 mutant background, DM fluke has compromised folding. Further, we want to see the motility of the worms in HSF1 DAP16 mutant worms. We did motility assay and thrashing assay. For motility assay, we observed the movement of worms at different time points and could observe that DAP16 mutant worms were moving freely whereas HSF1 mutant worms were paralyzed. Also, we measured the thrashing when we put the worms in liquid buffer and could see the movement of the tail and could find the DAP16 mutants were able to thrash better when compared to HSF1 mutants. This result show that HSF1 deletion in neurons can lead to paralysis of worms. Further, as a first step, we took the wild type worms and did a proteomics-based study to find out the key interactors of DM fluke in control and heat stressed conditions. We took GFP alone as a control so that we will be able to negate any interacting partner that may interact with only GFP. This figure shows the experimental plan where we took control worms and heat stressed worms. For control, we took DM fluke, no heat shock, and GFP no heat shock. And for stressed, we took DM fluke heat stressed and GFP heat stressed worms. Then we lysed the worms and did a co-immunoprecipitation with anti-GFP bits and followed by a mass spectrometric protein identification. Then we did the data normalization, cleaning, and filtering in Perseus, and we identified around 2,400 proteins, which we filtered based on minimum 3 valued values in at least one group. Then we did a Venn diagram, and here we could see that in DM fluke, there are 2,394 proteins interacting, and in DM fluke, no heat shock, there are 2,366 proteins, and there are GFP heat shock, there are 2,221 proteins, and GFP node, there are 50 proteins. And there are 1,973, those are common in all the 4 conditions. Then we were more interested to find out the interactors of DM fluke under heat stress condition. So we took different strategies to find out the interactors of DM fluke under heat stress conditions. So and also we did some analysis to find out the interactors of DM fluke in no heat stress conditions. So for this presentation, I will tell you about 2 strategies where we could find out only unique interactors. Those are interacting with DM fluke GFP at heat stress conditions. So there is a strategy B and strategy C. So here we compare DM fluke GFP heat stress with DM fluke no heat stress. And in the strategy C, we compared DM fluke heat stress with GFP heat stress. So, and we took log 2 fold change greater than 1 as a threshold for finding the enriched proteins in both the conditions. So if you look at the strategy B, we found out 108 proteins, those are enriched. And we find that 108 proteins are interacting with the DM fluke with heat stress. In the strategy C, when you compare to GFP, we found 240 proteins getting enriched, and they might interact with the DM fluke with heat stress. So then from both this strategy, we found we could find that HSP16s, those are small heat shock proteins. They are the top interacting partners from both the analysis. Apart from that, we also observed that there are PTM proteins responsible for post-translational modification, which gets enriched. So we took HSP16s and one of the PTM protein for further functional characterization. So to give a brief introduction of about HSP16s, so they are a family of small heat shock proteins, size ranging from 12 to 43 kilodalton, They interact with misfolded proteins and harmful aggregates, helping to reduce the toxicity or facilitate their clearance from cells. There are 10 small hits of proteins in humans and 13 small HSPs in C. elegans, composing of HSP12s, HSP16s, HSP17, HSP25, and CYP1. In order to assess the role of HSP16 in the context of neuronal protein aggregation, we generated a deletion strain where then we checked for the aggregates with the help of fluorescence microscopy using a wild type and delta HSP16 background. In wild type forms, DM fluke aggregates were visible under heat stress and dissipated during recovery, as it was previously also observed. Surprisingly, in the delta HSP16 strain, fewer or no aggregates were formed under heat stress, contrary to our expectation of increased aggregation due to impaired proteostasis. This suggests that HSP16s are required to form aggregates. Additionally, we conducted thermo-recovery experiments where worms were subjected to 8 hours of heat stress followed by recovery phase at 20 degrees. In wild-type worms, the aggregates formed during heat shock after 1 hour, whereas in delta HSP16, there were no aggregates. But when we give persistent heat stress to both the strains, we could see formation of aggregates. And when we checked for the recovery after 12 hours, the wild type worms efficiently recovering compared to delta HSP16 strain. In order to assess the role of HSP16 in stress resilience, we conducted thermotolerance experiments at day 1 and day 6 of adulthood, where we kept the worms in 35 degrees Celsius for their complete death. So in both day 1 and day 6, we could observe that DM fluke worms with HSP16 deletion shows less thermotolerant capacity, and with age, there is dramatic decline in survival under heat stress in the absence of HSP16s. So from our proteomic analysis, we also found that there are various PTM proteins enriched with DM fluke. So PTMs play a significant role in modulating protein aggregation, which is crucial for both normal cellular functions and in the pathology of various neurodegenerative diseases. So out of it, we chose OGT1, which is one of the interacting partner with DM fluke. From the proteomic study, we found out that it has a role in modulating protein aggregation. So OGT1 is responsible for a PTM called O-glucan-acylation, which is a nutrient and stress responsive PTM that involves the attachment of O-linked N-acetyl glucosamine moieties to serine and threonine residues of cytoplasmic, nuclear, and mitochondrial proteins. And OGA1 is another protein which is responsible for the removing of the O-glucanac moiety. First, we tried to find out the site-specific O-glucan-acylation in serine and threonines of luciferase protein through a bioinformatic tool, which gives the prediction of its site specificity. The threonines are in blue color and serines are in red color. We found 24 threonine sites and predicted to be O-glucanylated, whereas 29 serine sites were predicted to be O-glucanylated. The star mark in serine and or threonine show the sites where O-glucanylation and phosphorylation can both take place, where there is a crosstalk between O-glucan-acylation and phosphorylation. Then we check if the O-glucanac potential, where even if we put a strong cutoff of 0.5, we could find there are at least 4 sites where it can be O-glucanated in the luciferase protein. Further, to experimental validate it, we generated a strain where OGT1 is deleted in the DM fluke background and checked for the aggregates using fluorescence microscopy in the wild type and OGT1 deletion. In the wild type background, like previously, we observed with heat shock, DM fluke form aggregates and it get recovered when we put the worms back into 20 degree for 6 hours. But in the OGT1 mutants, with heat shock, we observe even more aggregates in the when compared to the wild type worms. And when we put the worms back into the recovery, in the OGT deletion, they were not recovering properly as compared to the wild type worms. We suggest that OGT1 is responsible for recovering heat-induced aggregates. So to further experiments are still undergoing to support these initial findings. So I'll summarize my talk. So we found HSF1, which can function as a disaggregate, and HSF1 DAF16, they functions differently. So HSP16s, which can help in formation of aggregates, that can be sometimes protective. And there are also PTMs, and we identified OGT1, which helps in recovery of aggregates. So the conclusion is HSF1 along with identified interactors may contribute to the regulation of neuronal proteostasis by modulating the folding and stability of aggregated proteins, potentially influencing the neuronal response to stress. With this, I would like to thank my supervisor, Dr. Prasad Kasturi, all the doctoral committee members, my current lab members, and the past lab members. So I'll thank Rusali, who helps me in performing most of the experiments, and hope to get some valuable suggestions and feedbacks from the mentors. Thank you.
Hi, very nice, very interesting work. I don't know a lot about the system, but my question is, so I assume that other proteins also undergo some sort of aggregation in response to heat stress. So I was
Yes.
wondering, what is sort of the ratio of proteins that you find that are aggregated when you look at these aggregates? Like, how many other types of proteins do you find in them? And are there some proteins which are more sensitive to heat stress and some which are not? And yeah, so that's sort of the first one.
So there are proteins with heat stress, they forms aggregates, and that ratio may vary. What kind of heat stress, and in the strain specific also, they are specific to specific strains. And as already aggregation prone protein is there inside the C. elegans, so there are chances that with heat stress, there are other proteins, those can co-aggregates with the this DM fluke double
Right.
mutant firefly luciferase. And the ratio, actually, it is hard to say. And exactly how many proteins are getting aggregated in the C. elegans.
And so you had this very nice piece of data where you gave heat stress at different times, right? So one
Yes.
was, I think, in day 1, and one was in, I think, day 6, and you're
Day
measuring
6,
the survival.
yes.
So clearly day 6, getting a heat shock at day 6 is worse than it is at day 1. So a couple of questions based on that. So post this heat stress, do levels of HSP16 change? And is there an alteration in how those levels increase depending upon the age of the animal?
So it's very, very interesting question. So the basal level of HSP16s, they increase with age. And when we give heat stress, anyway, they increase. But when we give heat stress at day 6, as already there are increase in HSP16s, and when we give heat stress at day 6, they their abundance still increase more as compared to the day 1.
No, because this survival curve seems to completely shift. Just looking at the wild type, not even looking at your various mutants, but compared to the top one to the bottom one, it's just totally different. So
yeah, in the wild type, C. elegans, when we knock out HSP16 in the wild type, there was no we could not see any change in the thermotolerance. But when there is a protein aggregate in the neuron, and there in that system we deleted HSP16, we found dramatic decline in the thermotolerance in both day 1 and day 6. But in the days as the worms ages, the thermotolerance capacity still decreases more.
And the same thing when it's
Just
done.
a minute. How do you say there's no difference in day 1 and day 6 in wild type? There seems to be a difference.
The curve looks complete. One is like this and one is like this.
And it, sort of everything is, sort of dies by 15 hours. I mean, maybe 50% is similar, but the shape of the curve is very different.
Yes, that day 6, we are still trying to replicate it. And this is the initial piece of work only. Means only duplicates we have performed, and one more time we have to still do it. And then we can tell a conclusion about the how it is decreasing in the delta HSP16 and DM flow background in day 6.
And the OGT, mutant, does that also show similar?
So that OGT one is a
not
very
done the survival yet. Okay.
Yeah, is a very preliminary one. And what we observed is in the when we delete OGT, their worms were not able to recover. And by looking at the aggregates also, we could find that there were increase of protein aggregates when we give heat stress to the OGT deletion worms when compared to the wild type.
Got it. Over to you, Sandhya.
So I have a couple of questions. One is that you have this marker protein that you're using for monitoring aggregation.
Yes.
Which you have expressed only in neurons. Is that, do you know if that is a neuron specific effect of aggregation, or do you see in your mutants, none of which are neuron specific, right? They're expressed in many tissues, that this effect that you're seeing, which is of your thermotolerance, etc., is actually applicable to all cells?
So these HSP16s, they are tissue specific, and they express in various tissues. But what we wanted to know that we deleted HSP16s completely, and now we wanted to check their aggregation potential.
This is on HSP16s are gone, right? This is not just neuronal one.
Yeah, the complete HSP16 we deleted from all the tissues.
Yeah. So have you thought of doing it? I mean, so you would expect these phenotypes to be in all cells or only in neurons? This aggregation, proteostasis.
So one more thing. So here we could see that when we delete HSP16, they were not forming any DM flow aggregates. So similar thing we also observed one more tissue in the muscle also. We saw the same pattern. When we deleted HSP16, we could not find any aggregates or very few aggregates in the muscle.
So the other mutant that you looked at was DAF16, right?
Yes.
And with DAF16, one of the things that's affected is aging, as I understand it.
Yeah.
Have you looked at your HSP and HSF mutants to see if the effects in aging? So the question I'm asking is, how much of it, so HSPs could be acting as chaperones to help fold proteins and unfold proteins.
Yes.
But the DAP16 pathway could be acting through an insulin signaling mechanism, which is intrinsically linked to lifespan. So have you looked at two things, the relationship between DAF16 and HSF or HSP to show that they're independent pathways? And the second is to see that the effects that you're seeing with DAF16, for instance, are not due to its aging-related insulin signaling pathway effects.
So first of all, I would like to say that whatever experiments we have done in this, so it's all in the early adulthood, day 1. And as you mentioned that there is the relation between HSPs and HSF1 and DAP16. And I would like to say that HSPs can be regulated by both DAP16 and HSF1. So, but that we are still don't know that by which DAP16 or HSF1 these HSPs when getting upregulated when there is a protein misfolding. So for that we have planned proteomic based approach again in these 3 mutant backgrounds, in the wild type, in the DAP16 mutant, and the HSF1 mutant. So similar to this, Similar to what I have shown here, we will do a immunoprecipitation with GFP, and we will.
Why not looked at other mutants which phenocopy DAF16 in aging phenotypes? Because that's insulin signaling in those animals is going to change from get-go. You may finally get animals which are long-lived and then make the case that it is really not insulin signaling, but it is DAF16. Wouldn't that be a equally valid approach? And then you could just make double mutants between DAF16 and HSF1 to sort of see how that changes. That might be faster and easier to establish.
Yeah, that is our just next plan to double mutant, to make a double mutant of DAF16 and HSF1 and to check for the aggregation dynamics.
But I would also check lifespan of HSF mutants. I would also check if age 1 and other kinds of mutants in the insulin signaling pathway show some of the same phenotypes. I think that could actually prove to be useful.
Yeah, surely we will do this, whatever you have suggested in these mutants also.
Sorry, I don't have anything else to say. Thank you. Nice
Thank
to
you so much. Thank you.
Thank you, Prasnadipta. You can answer a few questions in the chat window also. We will move to our last talk, which is by Kalyani. Kalyani, can you hear us?
Yeah, can you hear me?
Yes. We are able to hear you and we are able to see you.
Yeah, so you can see my presentation, right?
Yeah,
yes,
it is visible.
Yeah.
Also, Kalyani, please use point.
Yeah. The arrow is visible, right?
There, it would be.
Yeah, okay. So good afternoon, everyone. Sorry for the technical glitch that happened on my end previously. I'm Kalyani, working under Dr. Abhijit at BRIC NIB Hyderabad, and we are working on this lesser-known parasite known as Toxoplasma gondii. And as a part of the PhD café presentation, I'll be discussing my PhD work titled Essential Role of RNA Capping Enzyme Triphosphatase in Toxoplasma gondii Transfer Stability and Survival. So Toxoplasma gondii is an obligate intracellular eukaryotic parasite that is capable of infecting all the warm-blooded nucleated cells. And the life cycle of this parasite is very complex. It has a broad host range, but felids are known to be the definitive host, wherein the sexual replication of the parasite takes place. And via oocyst shedding, they contaminate the food and water sources. Upon consumption of these contaminated food and water by intermediate hosts like cattle, pigs, sheep, the parasite basically infects them and and reaches the dead end host, which are the humans. In humans, the parasite in an immunocompetent individual, basically, the parasite switches its life cycle from an actively replicating stage, which is the tachyzoid, to a dormant cyst-like stage, which is the bradyzoid, and lives happily in the brain, heart, and also causes ocular toxoplasmosis. One of the main clinical manifestation of this disease in females is abortions, both in cattle as well as humans, and that is why this disease is of zoonotic importance. The life cycle basically completes itself again in the cat intestine when it ingests a rat which is infected by toxoplasma. The life cycle of this parasite has these three unique life cycle stages: the oocyst, the tachyzoid, which is actively replicating stage, hallmark of acute infection, and the bradyzoid, which is the cyst form of toxoplasma responsible for the chronic infection. So these two stages are basically interconvertible. In an immunocompetent individual, tachyzoids rapidly convert into bradyzoids and they stay latent, and that is how a human being stays asymptomatic for years without knowing that they are actually infected by toxoplasma. The five of the genes are actually uniquely expressed across each of these developmental stages. And that's how we hypothesize that this parasite has a robust transcriptional machinery. The transcription in toxoplasma is found to be following just-in-time concept. And there exists paucity in both numbers and variety of specific transcription factors relatively to the high number of protein coding genes that leaves the possibility of alternate mechanisms open. But the post-transcriptional level of regulation in epicomplexin, including toxoplasma, seem to be held by very complex mechanisms, and most of them still remain understudied. mRNA capping is one such modification that occurs at the 5' end of RNA on the nascent RNA, which is transcribed by the RNA polymerase II. Coming to the process of mRNA capping, it is a sequential process and differs between the lower and the higher eukaryotes. So we have in the schematic, we have the RNA polymerase II actively transcribing mRNA, which is inherently triphosphate ended at its 5' end. The first protein, RNA triphosphatase, comes up and cleaves off the gamma phosphate. The second protein, which is the RNA guanylate transferase, detects this diphosphate-ended RNA substrate, and then using GTP as a cofactor, cleaves it and adds the GMP group, again making it a triphosphate-ended RNA. One point here to note is, in the lower eukaryotes, there exist two different enzymes, the RNA triphosphatase and guanylate transferase, whereas in the metazoans, which is humans and plants, these two enzymes exist as a bifunctional product, wherein the N-terminus is RNA triphosphatase and the C-terminus is guanylate transferase. The third enzyme, which is the methyl transferase, uses S-adenosyl methionine, SAM, as a cofactor in order to methylate the N7 position of guanosine, and that's how we have the M7G capped RNA. It is important to note that viruses have evolved with a highly diverse capping mechanism. Either they cap snatch the RNAs from the cytoplasm and use it as a primer for their RNA-dependent RNA polymerases, or they have a complete different mechanism in which all these three proteins are present in tandem. So on the basis of this background, we have formulated three objectives for the study, which includes identification of Toxoplasma gondii RNA capping enzymes, their characterization, and to study their role in productive mRNA synthesis and gene expression in the parasite. As of now, in epicomplexins, leave apart Toxoplasma, there exists just one report in Plasmodium that tells the existence of these enzymes, and we are the second one to work on this and report it for the first time in Toxoplasma. So to begin with, we basically went ahead using the M7G commercial antibody and did a IFA in both sexual stages of the parasite, the tachyzoid as well as the bradyzoid, and we could see a clear localization of M7G signal in both the stages. Then we isolated parasite RNA and went ahead to do a dot blot. And as you can see, the dot blot is also positive and shows a good M7G signal. Then using BLASTP searches, using the yeast and Plasmodium as a query, we could identify three separate enzymes in Toxoplasma as well, and name them as TGRT, TGGT, and TGGMT. Upon mining the literature, we could see that there exists a clear difference in the organization of these enzymes when you compare them from metazoan to epicomplexin. In the humans and plants, there exists a bifunctional RNA triphosphatase and guanylate transferase and a separate methyl transferase. In fungi, there exist three separate enzymes. In ciliates, it is important to note that they have an organization similar to the higher eukaryotes, whereas kinetoplasts have a single triphosphatase and a fused second and third enzyme. What we could see in Toxoplasma is that it is a little similar to the fungi group, wherein you have three separate enzymes. So this is a schematic representation of these three enzymes, wherein triphosphatase is a 920 amino acid protein, guanylate transferase is a 509 amino acid protein, and methyl transferase is a 1283 amino acid protein, which is an exceptionally the biggest of all present in the epicomplexan. So this is an observation which we have made, but we could not assign any specific roles to these extended terminal for these proteins. We have focused just on their function in our study. In order to be sure that what we have identified is indeed the mRNA capping machinery, we went ahead to do yeast complementation assays in which these three proteins are individually genetically knocked out in the yeast strain, and using yeast shuttle vector, we basically cloned and transformed these individual deleted mutant strains, and we were able to restore the growth of these mutant strain. And then finally, as a proof of concept, we even procured this triple deleted mutant wherein all these three enzymes were knocked out, and doing a co-transfection with these three vectors, we could see that the growth is restored, which means that these three identified putative proteins are actually mRNA capping proteins. One thing to note here is in this gene name followed by CRISPR score, that this score basically specifies that these three enzymes are highly essential for the parasite, and knocking out any of them would be lethal for the parasite. So this background would actually help me to explain my slide further. So next, that we have identified these enzymes, it was important for us to characterize them and see whether they're active or not. So we cloned express purified full-length triphosphatase protein, and since there are no commercial antibodies available for this parasite, we went ahead on to generate our own polyclonal antibodies in mouse, use them to see the localization of this protein, and as you can see, it is expressed in both tachyzoid and bradyzoid stages with a prominent nuclear localization. Then we went ahead to do a triphosphatase assay, mentioning in my introduction slide that the triphosphate RNA acts as a substrate for the first enzyme. So here ATP, which was used as a substrate, was gamma phosphate labeled, and if the protein would have been active, you would expect the release of the inorganic phosphate, which would be captured through thin layer chromatography. So here in D part, we have a schematic, we have a representative result wherein we could see that this enzyme is active solely in the presence of manganese and not in the presence of magnesium. So as I had mentioned in my previous slide, that these are metalloenzymes in lower eukaryotes, and the same enzymes have a different structure and a different mechanism of action in higher eukaryotes, including humans, and they belong to the human cysteine phosphatase type of family. So here we basically prove that this enzyme is indeed a metalloenzyme, and also quantified the PI release at different time points. We were surprised to see that this has a pH flat profile, meaning that it's actually active across a wide range of pH. Then we did some structural studies using AlphaFold and CoLabFold. Since this protein has a really less homology, just 22% with the yeast counterpart, we were able to identify a few residues which are actually conserved in the metalloenzyme triphosphate tunnel type proteins, TTMs. So we went ahead to generate two different mutants of such identified glutamate motifs at E366 and E726, mutated it through alanine using site-directed mutagenesis, and went ahead to check the activity. And as you can see, the mutated proteins show a reduced activity. All these results collectively establish that TGRT is indeed a triphosphate tunnel metalloenzyme unique to the parasite and is mechanistically and structurally different from the human counterpart. Going on to the second enzyme, which is a guanylate transferase, we adopted the similar method, raised antibodies, showed its localization in both of the stages, and went ahead to do the guanylate transferase assay, wherein the alpha phosphate was labeled. So if the protein is active, one would expect that the inorganic diphosphate is released without any label, whereas the GMP moiety stays back with the alpha phosphate being labeled. We have captured this through autoradiography in D and E. And here again, the guanylate transferase was found to be active only in the presence of MnCl2. One peculiar thing to tell you here is that this protein follows the canonical process of guanylin transferase activity as in humans and higher eukaryotes. And it has this KTDG motif wherein the lysine of at this is a okay, so this is a motif which is responsible for making a nucleophilic attack at the GTP. So with SDM approach, we went ahead deleting each of this protein, replacing it with alanine, and we could see a complete abolished activity of this protein, which means that it has a canonical role and has this KTDG motif, which is important for its functionality. Now, getting substrates for the first and the second enzyme to show its activity was simple. But when we went ahead to characterize the third protein, which is actually 150 kilodalton protein, we decided on to just simply clone and express the C-terminus part of it, because the N-terminal region did not have any SAM binding motif. So we expressed the partial protein and successfully showed its localization in both the stages and generated the reagents, basically the substrate for this protein in-house. So what we did was we have this T3 promoter that transcribes 32 more RNA. We went ahead with in vitro transcription IVT using T3 polymerase and subjected it to RNA triphosphatase and guanylin transferase along with GTP in the reaction. And this guanylated product was then taken up for the methyltransferase assay. And we again spotted these reactions on the membrane and probed it with M7G commercial antibody. Vaccinia capping enzyme VCE was used as a positive control, which we got through NAB. And as you can see, we have the third protein whose C-terminal part is also functionally active, as it was able to cap the guanylated RNA. In order to be specific, that to be sure that SAM is the cofactor used, we used a structural analog of S-adenosylmethionine, which is sinefungin, performed a similar experiment with the inhibitor, and we could see that indeed the activity of the enzyme is being inhibited. Okay, so next now that we had the capped RNAs in hand, the protocol in hand in the lab, we wanted to see whether or not these capped RNA interact with the cap binding protein. So we all know that elongation initiation factor is a group of proteins which are present in the cytoplasm and they bind to the M7G cap RNA. So for the first time, we identified this Toxoplasma gondii EIF41 protein and showed its presence in the cytoplasm of the parasite. And then we went ahead with the same strategy of generating the 32 more RNA, capping it in the lab, and having an interaction assay using microscale thermophoresis, MST assay, in the tube, wherein we labeled the his tag TGAF4E with the commercially available kit and titrated the analyte, which is the capped RNA, against the labeled protein. So here are two controls wherein the triphosphate RNA alone and the guanylated RNA alone was not able to interact with EIF4E at all, whereas the capped RNA M7GPPN32 was able to interact with TGIF4E with the nanomolar range, which means that it's a tight association and a high affinity interaction. This basically proves that the capped RNA generated in lab can actually be used for interaction with the elongation initiation factor. In order to take this further in the in vivo scenario, what we did was we made a construct wherein the T3 promoter expressed the firefly luciferase. So in the first condition, we had the triphosphate ended RNA alone. In the second condition, we had the capped product, whereas in the third condition, we had the capped as well as the poly A product, the RNA product. So we went ahead with RNA transfection and quantified the relative intensity of luciferase. So both in vitro and in vivo scenario, now we are sure that the capped RNA which are made in-house using these proteins are actually able to be sensed by Toxoplasma gondii EIF4E. Now, having mentioned that the first protein is unique to the parasite, we wanted to dig out the role of this protein further. So since this protein is refractory to knockout due to its negative CRISPR score, we decided to go ahead with knockdown of Toxoplasma gondii RNA triphosphatase. So the strategy which we have used here is an auxin-inducible degron system, wherein we have a parasite line expressing TIR1, the tachizoid form, the RH strain, and when we have the protein of interest tagged with MAID cassette, followed by HA for localization. When we transfect up, we do transfection using this strategy, wherein we have the promoter for RNA triphosphatase and the 3' UTR. Using CRISPR-Cas9 based approach, we basically flip apart the last region of the gene followed by the UTR region in order to incorporate the AID cassette, the HA tag and HXGPRT for the selection, and transfect this in the parasite. Wait for a couple of months in order to get the clonal population which is resistant to MPA and xanthine, and then to check whether or not it is working, we supplement it with auxin IAA. So what happens is the TIR1 takes up the auxin and induces the ubiquitination of the protein of interest, in this case RNA triphosphatase, which is tagged with auxin-inducible degron cassette, and there happens a total proteasomal degradation, and as less as 30 minutes we can see a complete depletion of this protein. So we went ahead with the strategy for the first protein since it was unique and we wanted to study its role in the parasite. We confirmed it through diagnostic PCR and Western blot that in as less as 1 hour, there was absolutely no detectable level of TGRT in the parasite. We also confirmed it through immunofluorescence assay and performed a dot blot at different time points, of which we could see that at 8 hours, there is a reduction in the M7G levels in the parasite, which we have quantified here. Next, we went ahead to do a luciferase-based reporter assay, in which there exists no specific assay in order to see how in real time or how in vivo scenario the absence of capping is actually affecting the parasite or the translation of a protein.
Kalyani, sorry to interrupt.
Yeah.
Kalyani, 20 minutes are up, so you should start wrapping up now.
Yeah. Sure. So basically, we did a few phenotypic assays in order to show that the replication of the parasite is getting affected, and the parasite fails to form plaque, which means that there is a compromised infection when you have the deletion of this protein. So in order to see at the molecular level, what are the changes which are happening, we went ahead to do RNA cap sequencing, wherein this strategy was followed, and we could come up with certain molecules, a certain group which were actually affected. So the histones and the proteins which are responsible for maintaining the nuclear integrity and structural integrity of the parasite were found to be highly dysregulated, specifically downregulated in this case. That explains why the replication of parasite was halted. As the final proof of concept, we went ahead to do the animal experiment, wherein we have a control group and an IA group. The control group and both the groups were basically infected with Toxoplasma gondii. The IA group was fed IA through water regularly, and when we isolated PECs, we could see that there is no protein. There are parasites, but absence of the RT protein, and they showed a full survival, 100% survival when you do not have the triphosphatase. So we then went ahead to see the structural conservation of these RNA triphosphatases across different group of organisms. This is the one in a parasite, which superimposes with the yeast. The Trypanosoma brucei CET1 is also being characterized, and there exists certain level of identity with the human triphosphatase. So the humans encode only one single triphosphatase, which is the thymine triphosphatase. But due to the presence of this coil, as you can see, the substrate specificity is maintained. So this triphosphatase cannot actually work on the RNA. So to summarize this work, and especially the RNA triphosphatase work, what we have here is the normal parasite growth when you have a productive mRNA capping in the presence of normal TGRT, and a normal growth of parasite in the in vivo scenario leading to lethal toxoplasmosis and death of the mice. But when you have a IA-induced TGRT knockdown in both in vitro and in vivo scenario, there is arrested parasite growth in the culture, and the mice survive happily as these parasites are unable to have a successful infection. So we can extend this study, and the E part basically indicates the possibility that smaller molecular inhibitor that specifically target the TGRT tunnel can be developed, so that we can have a very specific target for this parasite. And yeah, that's all I have to present. I would like to acknowledge Brick, NIB, my supervisor, all the lab members, especially my senior Ms. Poonam. I would like to acknowledge DBT, Manipal, and India Bioscience. And thank you for your attention.
Okay, we can get to some questions, Sandhya and Deepa.
Yeah.
Thank you, Kalyani.
Thank you.
Kalyani, can you go back to your previous slide?
Yeah.
Actually, one before this, where you had the overlap. No, the overlap structures. Does that come from AlphaFold?
Yeah.
Okay. So, do you think that, you propose this mechanism in the end that you could get inhibitors which are specific for the toxoplasma, but may not act on the vertebrate species? Have you tried? I don't know if this experiment is possible in toxoplasma, because I know it's not a standard model organisms. Have you tried to supplement the human gene in toxoplasma to see whether it can supplement those effects, to see whether the other partners that it works with are actually different? So even if you don't get, because the structure to me looks there's quite a bit of similarity between the human and the toxoplasma structure, but there could be accessory proteins or accessory interactions which are much more important. But for that, your human protein should not be able to really function well in toxoplasma, or that's what I would have thought. But anyway, I'd be curious to know what you think.
Yeah. So the first thing is, this protein is not the capping enzyme of humans. It's a thymine triphosphatase, and it has been extensively characterized in mouse and humans that this enzyme solely uses thymine triphosphate as a substrate. And the substrate specificity here is ensured because of that coil region, which you can see as an extension present only in the red protein. Secondly, the mechanism of action of toxoplasma RNA triphosphatase differs completely from that in the human. So in the human, there is this HCCX motif which is present, the cysteine being the catalytic motif here in humans. Whereas in toxoplasma, it encodes these glutamins, which I had shown in the mutation data. These glutamin motifs are responsible for coordinating the Mg, Mn, and water molecule in a way that the RNA substrate is identified. So I think these proteins are structurally, mechanistically totally different. The purpose of putting up this red protein at the end was just to show that there is only one single triphosphatase in humans. That is also not a nucleotide triphosphatase. And to rule out that possibility as well, I have done the superimposition.
So in a sense that the superimposition is, you do not expect, let's put it like that, you do not expect knockdowns of this gene where if you target these, you're going to have some sort of cross reactivity with any of the other genes in humans. Is that your based on that?
Yeah.
Okay. All right. Thanks.
So toxoplasma also doesn't always seem to cause lethality or disease, right? So there seem to be conditions where it's present in an organism but does not really manifest, any phenotype.
Yes.
I was wondering, is anything known about this latency and not displaying a phenotype, and how does that correlate with your TGRT levels? because your treatment with I hear, your depletion of TGRT seems to have a very drastic effect. So I was wondering if this is something this sort of information can be got from people infected with toxoplasma and don't display, any disease phenotypes.
Yeah. So the reason why we have asymptomatic nature is because of three different strains of this parasite. The first strain is the is highly virulent RH strain. The second strain is ME49, which is mildly virulent and is capable of transitioning to bradyzoids, the tissue cyst. That makes its latency possible. So for the first enzyme, the LD is 1. So even if you're infected with one parasite, you're bound to suffer from neither toxoplasmosis. But in case you're infected with a second strain, these parasites can convert into bradyzoids and stay inside. So that's how most of the human population, almost 1/3 of the human population, is positive for toxoplasma, but still stays unaware because they are asymptomatic. And as far as the experiment which you have performed is in RH strain, because the TIR1 line in which we have made this conditional knockdown has a RH background. And we even collected the bleed from these surviving mice and did PCR for toxoplasma specific genes after day 30, but we could not find any positive PCR for it. So even we did a bradyzoid marker specific PCR, which was negative, which means that there was indeed a clearance of these parasites when we supplemented the mouse to water with IAA.
But across the three strains that you mentioned, the less virulent, the more virulent, and the super virulent, there's no difference in TGRT levels or expression?
So the information which we have on the toxoplasma database at TUPAT-DB is on the basis of ME49, the type 2 strain, which is well studied. And we could, there exists no change as much as the mass spectrometric levels are concerned. The peptide level, we could not see any change in all these three strains.
I see.
Yeah.
And the other two enzymes, so initially you mentioned that there are three enzymes which are present in Toxoplasma gondii. One is specific to the parasite and the other two seem to have some similarity to other. So what is known about the individual functions of those two and what happens when you deplete them? Have there been studies in the past?
So this is the first study which has been done targeting a mRNA capping enzyme in the entire eukaryotic system. There has been a study in communications biology in January 2024, wherein they have depleted East guanylin transferase, the second protein. Okay, and what they have shown is that when you deplete the second protein, there is no change in the RNA polymerase II occupancy. So how these enzymes function is by using the RNA pol to CTD as a launching pad. So you have interactions of these enzymes with them, and that's how these enzymes know that the RNA substrate is present and they just go and cap because there is no sequence specificity. So one study has been done using yeast model, and as far as methyltransferases are concerned, there are quite a couple of studies using the human and the mouse model, wherein they show two isoforms of methyltransferase, one in the cytoplasm and one in the nucleus. So there is also this concept of recapping that occurs in the metazones. But as far as knockdown is concerned, no. This is the first study of its kind.
Cool. Thanks a lot.
Yeah.
Thank you, Sandhya, Deepa, and Kalyani. That was an exhaustive, intensive session. I will share my screen and this time not forget to give out the name of the giveaway winner. And the giveaway winner is Sanjana Sarkar. So congratulations, Sanjana, and send us an email at hello@indiabioscience.org and we will ship your physical copy of this newly released e-compendium, hot off the press, right? This is what we call hot off the press. So we had a very good open Q&A, and audience, we have dropped the feedback form multiple times. Please give us your feedback. We really want to see how we can maybe take PhD Cafe further. So thank you all. Thank you, Deepa, Sandhya, all our speakers, our audience. Tomorrow morning, 10 a.m., neurobiology. So be there. All right. We have again a lineup of 5 speakers, and tomorrow afternoon is microbiology with a lineup of 6 speakers, and we have an open networking session for 1 hour in between that. But don't worry about the details. Just show up tomorrow morning at 10 a.m. for neurobiology. All right. So thank you once again, Sandhya, Deepa. A lot of very, very good feedback, lots of good questions. Thank you. I hope you enjoyed it.
Thanks a lot. It was
Great.
amazing.
Great.
It was very nice.
Thank you so much. Yeah.
Thank you for asking us to
Thank
come.
you. To think of all the good work happening at PhD level in India. Very exciting.
Yeah.
Yes. Thank you so much. And we will see you tomorrow morning, the rest of you. Take care. Have a good evening. Bye-bye.
Bye.